All right, maybe we should get started. Welcome everyone to the first of our symposia for this academic year. Like everyone, we of course had hopes and fantasies of all being in person, which didn't come true for reasons that you all know, but I'm happy that so many of you have the energy to join us via Zoom once more. So today is about the physics of behavior, which is really a field that's been incredibly lively over the last decade or more, and has undergone really dramatic changes. And that's happened in terms of the investigation of the behavior of individual organisms, but also the investigation of the behavior of large groups of organisms, and also descending or probing the neural basis of these behaviors. And so we're going to touch on all of these things today with our three speakers. I'll remind you of the format. Each speaker has a block of an hour and a half. To make things livable on Zoom, we'll then just have a half-hour break. You can get up, walk around, get yourself a glass of water or a sandwich, whatever you need to do. In person, these events tend to be very interactive. People interrupt with questions and so on. I think what Orit is planning on doing is sort of pausing twice during the hour and a half for questions at roughly equal spacing. If there's something that ordinarily you'd interrupt because it's really not clear, why don't you put it in the chat and I'll monitor that and do the interrupting. And then afterwards, when it's time for questions, I think the easiest is if people unmute, but we'll see how it goes. Also, please feel free to use the chat, and I will keep an eye on it if questions are accumulating. So with that preface, let me introduce our first speaker, Orit Peleg, who's coming to us, well, physically from Israel at the moment, but she's on the faculty at the University of Colorado in Boulder. Has worked on a variety of problems in collective animal behavior, particularly with insect swarms, and she's going to give us a sampling of this. You'll notice that the picture credit is in part from National Geographic, so you get the sense that her work has attracted some attention, as it should. It's been very beautiful and insightful, and in particular has gone off to look at new and different systems that haven't been the ones that everybody else has been studying, and that's always very exciting. You're going to hear from her about the science. I wanted to add one thing, and actually it's interesting that today all three of our speakers are people who have spent quite a lot of energy on teaching and education in its many forms, and I wanted to draw attention to something that many of you will know about, which is the presence of the American Physical Society's Division of Biological Physics in social media, and also in a series of regular talks and discussions, digests of work, and so on. And Orit has been one of the people who's really led this effort, which I think has been an enormous service to our community, especially in the time of the pandemic, but I hope that it will persist beyond the constraints that we've had for the last year. So, Orit, please.
Okay, thank you so much, Bill, for the lovely introduction. Can you just confirm that you can hear me?
Yes, I can, but then
awesome.
I'll mute. I can hear you, but now I'll mute myself.
Okay, sounds good. Thank you so much for the introduction, and thank you so much for inviting me. It's really exciting to join you from very far along with the globe, and tell you a little bit about some of the work I've been doing over the last couple of years. It's going to be focused on physical computation in insect swarms. But before I dive in, I thought we'd just take a step back and tell you a little bit more about broadly what's defining the questions that are driving research in my lab. I tend to think a lot about animal communication in nature. And as I'm sure all of you are well aware, especially now in the times of the pandemic, we sort of all got used to being able to communicate with each other over large distances. We have mobile connectivity and a bunch of backup systems like mail and satellites and so on. But of course, animals have to solve similar challenges only with the natural resources that they have at their disposal. And there's a beautiful variety of communication in nature, in pairs of organisms, for example, tactile communication, visual, chemical, vocal, and any combination of these modalities. But animals didn't just evolve ways to communicate with each other in pairs, they also can do this in groups, and it can also happen in very large groups. And this is the field of collective behavior and communication in collective behavior, which, as Bill already mentioned, has been sort of on the rise over the last couple of years or a decade. And I just want to mention, briefly highlight here two examples of this collective communication. One of them is shown here in the movie at the top right. And this is an experiment done with slime mold. All these white dots that you're seeing here on the screen are food sources. And then the slime mold is going to use chemical communication to recruit other parts of the superorganism to the food sources. And in a sense, they also find the shortest path in between all of these food sources. For those of you who are familiar with this experiment, it also happens to be the reconstruction of the transportation map in Japan. And something about the ways that the slime mold solved this problem is actually more efficient than what humans did in order to design these transportation networks. So I'm mentioning this because, in a sense, the slime mold is performing some kind of physical computation. In that case, it's the nearest path between food sources. And then the other example I wanted to mention is these beautiful flocks of birds, of starlings, that had been shown to use visual communication to coordinate their movement in these really, really large groups and move effectively and efficiently in make these sharp turns and so on. The kind of questions that I think about a lot are, how do these communication signals in the context of large animal groups are generated and how they are interpreted? We can think about detectability. So how can one increase the signal to noise ratio, compression, so minimization of information or minimization of energy, robustness, so how sensitive everything is to the failure of individual animals or individual nodes. And the approach in my lab is mainly to design testable phenomenological theories. It's really, the emphasis is on testable theories that we can actually then test with real animal systems, and sort of go back and forth between predictions that the model make to experiments. And I think this is maybe a good point to just clarify upfront that the mathematical models or computational models that I will show in this talk, I'm not going to claim that this is really what the animals are doing, because of course we, at least so far, don't have the ability to really peek into the brains and record on a neural level what's going on in terms of the computation. But I want to make the point that these models are still useful because they're able to provide a mechanism, and a mechanism can make predictions. And predictions are useful because we can test them and then go back to the model if it needs any refinement. Okay, so this is the approach. And in terms of the behavioral experiments, work in my lab has been mostly focused on insect swarms with either fireflies or honeybees. And we work with these systems not just because they're so cute looking, at least in my opinion, but they really push the limit of communication in large groups in a way that is traceable. So by looking at the organism, we can say what kind of information to some extent is being communicated. Okay, so my plan for today is to highlight three projects that relate to communication in these large groups of insects. The first example would be mechanical communication. This is work that I started when I was a postdoc a few years ago, before I started my own lab. And I'm going to tell you about some of these living materials that the bees create by linking their own bodies. Next, I'm going to move to chemical communication, still in honeybees. And then I'm going to switch gears and talk about fireflies and visual communication with fireflies. And I think my general plan is to have short breaks for questions in between these three parts. But as Bill mentioned, please do raise any questions if you have them in the chat. Okay, so let's talk about honeybee mechanical swarms. So if you know me for a while, you probably already heard me talk about this, but I still find this problem very exciting, even though I started working on it a few years ago and still working on it in my lab. So I'll start with showing you some of the basics, and then I'll briefly highlight some of the new results that we acquire in my lab. Here on the left side is an example of a honeybee swarm. This is a behavior that occurs as part of the reproductive cycle of European honeybees. At some point during the spring or summer months, the colony size, the number of bees in the hive increases, and they split into two parts. And in one part, and the queen fly away and hang like this from a tree branch. From that point, they send scouts to find more permanent position for the nest, and they communicate that position using the waggle dance, which is a fascinating, another fascinating way of mechanical communication, which I won't have time to get into today. But the point that I want to make here is that this is a temporary structure. So the bees don't have a permanent nest structure that would potentially protect themselves from the elements. And because of that, the bees need to be able to adapt dynamically to environmental changes. One of the reasons that the bees create this structure in the first place is for thermoregulation purposes. So they huddle. By huddling, they reduce the average surface area per individual. So that helps their thermoregulation. And because of that, it has been already shown that the bees are able to adapt to environmental changes in the ambient temperature. So the top two figures here, these are taken from some beautiful work from Tom Seeley, who studied swarms in more natural environments. And this is the same swarm under different ambient temperatures. So on the right side, this is cold environment. So you can see that the swarm is, the volume of the swarm is smaller, which means the density is higher. This is what they do at cold temperatures. And then on the left side, this is the same swarm at warmer environment. At the bottom here, there is a picture of the swarm when it rains. And they tend to align their body axis along the direction of gravity. It's called, in the beekeeping jargon, shingle-like configuration, because it looks like roof shingles, and presumably make the swarm less susceptible to water. And then at very high temperatures and very cold temperatures, there's been some hypotheses about what the internal structure of the swarm is. And then at high temperatures, Bernd Heinrich speculated that there's channels that are basically empty of bees, and they're helping the bees to ventilate and exchange air from inside the warmer swarm to the colder environment. So these are this is a very impressive set of skills that the bees have in order to adapt to environmental changes. But underneath all of this, there is a difficult mechanical problem, because the bees must maintain mechanical stability. So one thing that I want to emphasize here is that the bees are not flying. It's not a flying swarm. As you can see, the bees are actually holding hands, in a sense, to create this very dense structure. And they're going to have to deal with a few types of mechanical forces. One of them is static, and it's just coming from gravity. And the combination of that and thermoregulation, and also the shape of the branch, will result in different shape of the swarm. So here are three examples of that. But then they also have to adapt to more dynamical mechanical perturbation, for example, coming from wind. So here at the movie at the top right, which I hope you can see playing, we have a swarm on a windy day. The branch is going to get shaken, therefore the swarm is going to get shaken. And then if the swarm breaks, this would basically kill the thermoregulation purpose of the structure. It's very important for the bees to maintain this mechanical stability, but it's not really clear how they manage to do this, mostly because the size of the swarm. So there's tens of thousands of individuals in each one of these swarms. I want to make a point with this little cartoon here, which is that a bee at one side of the swarm can potentially coordinate its activity with neighboring bees or maybe second neighboring bees, but there's going to be some finite distance beyond which they're not able to communicate with each other in this really dense environment. And certainly not communicate directly with a bee that is positioned at the completely other side of the swarm. So how do they maintain mechanical stability, which is a task that sometimes demands the simultaneous coordination throughout the different parts of the swarm? That was the question that we wanted to answer. And specifically, we wanted to understand what could be potential sources of local information that just around the vicinity of this individual bee, there's going to be some information that the bees must be using, and we want to know what it is in order to stabilize the structure of the swarm. Okay, so this was our field site to try to address that question. We worked with real honeybees, and we worked at the Concord Field Station, which is not far away from Harvard. And the setup was fairly frugal, partially because we didn't have a lot of money for funding this experiment, and partially because this was, at least for some of us, the first venture into experimental biology, I think you can say. So this was the setup. The swarm is attached underneath a wooden board, and that wooden board is controlled by a motor. And we have several cameras that record the swarm from a few different angles so that we can follow the shape of the swarm as a function of time. And what we're doing in experiments is simply shake the bees, kind of like this. In this movie, you're seeing an experiment where we're shaking them right and left. And we're following what the bees are doing as a function of time. And you can probably already see that the tip of the swarm is a little bit jiggly. So it's not a very stiff solid. There's going to be some elasticity that will play an important role in the response of the bees. So that was the experiment. We discovered that when we run the experiment for a longer duration of time, in this, what I'm going to show you here, it's for about 15 minutes, then the bees collectively change the shape of the swarm. And I want to show you a fast version of this experiment. So we have here the front view of the swarm and the bottom view, and we're shaking them right and left along the plane of the slide. And this is how our experiments look like. So the swarm starts from a cone-like structure that has a certain height and a certain base area, and then we're shaking them, and they're basically spreading out and decreasing the height and increasing the base area for the grip on the board, which makes a lot of sense. But still, we wanted to, sorry, I should also mention that here, just around here, we are stopping to shake them so we can also see how they slowly but surely go back to their steady state, so the more elongated swarm. So we studied this phenomena a little bit more systematically. For example, here we tested the effect of the amplitude of the acceleration that we apply on the board. So the y-axis is the relative base area of the swarm, the x-axis is time. Different colors correspond to different accelerations. And as you can see, the higher the acceleration is, the more the swarm spreads out. But then when we, instead of applying this perturbation and shaking the bees right and left, we shake them up and down, just the same swarm, the same kind of mechanical perturbation, we see a very different response. So again, the y-axis here is the relative base area. So we see that the swarm doesn't spread out. It doesn't respond in the same way. Even cranked up the acceleration here to test whether maybe higher forces are needed to get the bees to spread out in the vertical perturbation. But actually, after a certain threshold of acceleration, there's going to be at some point a crack that propagates through the swarm, and it's going to break, and that's very inconvenient for the experiments and for the bees. So we didn't really explore that regime too much. But still, there's a really interesting question to ask, which is why did the bees respond to horizontal perturbation but not to vertical perturbation, at least within this regime of accelerations? And to answer that question, we turn into a computational model, modeling the swarm as an elastic material. And we're going to look at the mechanical properties of these materials, of the swarms at different aspect ratios, starting from very elongated to very spread out. And the reason we do this is because the time scale in which we apply the mechanical perturbation, which is around the order of a second or a fraction of a second, is much smaller than the time scale that it takes the bees to spread out, which is closer to 30 minutes. Okay, so the model is a fairly simple representation of a bee. It's a spherical bee that is connected by elastic springs to its neighboring bees, and we arrange them like this in a cone-like structure. We have gravity, and we solve these equations of motion. We added the friction term here because we know from the experiments that if we shake the swarm once, it will sway kind of like a pendulum, but eventually it will stop swaying, and so there's obviously some friction involved. And we're going to look at a candidate source of mechanical information, and that is the stretching of bonds between neighboring bees. So for each bee in that simulation, we're going to look at the springs connections that it's making to its neighboring bees and measure how much they stretch as a function of time. And then just take the geometrical average of all of the connections per individual bee. So here is an illustration of how those simulations look like. So this is a three-dimensional swarm cut along the plane of this slide so that we can see what's happening inside. And the color here corresponds to that amount of local signal that I mentioned in the previous slide. So blue is low and yellow is high amount of stretching or shear in that case. And at the top movie that I'm playing now, this is a horizontal perturbation. There was quite a lot of deformation happening closer to the base of the swarm, which is where the perturbation is coming from. But then when we apply the same perturbation just vertically, we see that there is a very different amount of stretching of these bonds. And we can see the same kind of behavior in the experiments. So here, what I'm showing you is we cannot really track individual bees too well in these swarms, but if you look at a row of bees along the center of the swarm and follow the maximal amount of deformation between each layer along this line, we can see that indeed for horizontal perturbation, the amount of stretching, effective stretching between the layers in the swarm is higher than when we perform the experiments vertically. We can also look at the effect of aspect ratio. So here I'm showing you again the same swarm, the elongated swarm from the previous slide compared to a spread out swarm. And again, moving from this elongated configuration to the spread out one also effectively decreases the amount of stretching that these bonds are experiencing. And again, this is something that we can see in the experiments. So again, if we just follow one bee per layer and plot the propagation of these tracking as a function of time, we can see that for an elongated swarm, there's going to be more mechanical deformation in comparison to the case of a spread out swarm. Okay, so we have potentially a good candidate for a local mechanical rule or a local mechanical source of information that the bees might be following. That would explain our experiments. So now, just as a proof of concept, we took all of that information and extended their simulations to be agent-based simulations, where now the bees can actually sense these mechanical forces, and then if they reach a certain threshold, then they do something about it. And so here I'm showing you that mechanical stretching is shown with a color scale. So like before, blue is low and yellow is high. We already know that for horizontal perturbation, the amount of mechanical deformation is going to be higher in comparison to vertical. This is just a proof of concept, so I'm going to choose to set this threshold that triggers some behavior from the bees, such that vertical shaking is not going to get a lot of bees triggered. And then we just need to decide once the bees are triggered, what should we program them to do. So from the experiments, we know that the bees move upwards, both from tracking individual bees, but of course, this would lead to the spreading of the swarm. But we were not sure that individuals inside very shaky and condensed materials would know where upward is. So we decided to implement that rule based on more local information, and that is the gradient of the local mechanical signal that we defined. So that gradient is shown here in black arrows, which are a little bit small, but if you, hopefully you can see them. If not, they're really pointing upwards and a little bit to the sides. Okay, and then we just put everything together and run these simulations. And then perhaps not very surprisingly, again, this is really just a proof of concept. The simulations where we shake the bees horizontally are spreading out versus the case of a vertical perturbation.
So, Orit, sorry.
Okay. Yes. Yes.
Maybe you could go back for a moment. So I actually had a question. There's also a question in the chat. So a question in the chat was whether the holding hands that you described is happening both horizontally and vertically. So do they have any preference about how they connect?
Oh, yes. So that's, of course, we made this simplification assumption that we have a very regular lattice and isotropic in the sense that the connections in the simulation can occur with equal probability in all directions, but actually for the bees, that's not quite the case. So they have a preference to hold on to bees that are kind of in front of them, just because of the opening angles of their limbs, and not so much for bees that are behind them. So we see more holding of in front, and that can actually break the symmetry, in a sense that would totally disvalidate our model.
And I mean, is this an interesting direction to go, or?
Yeah, so I think it's definitely an interesting direction to go, and maybe I can, in a few slides, I was going to show some experiments that we do now with X-rays, where we still unable to see the breaking of symmetry in terms of in the directionality of the bonds, but we can see some breaking of symmetry in other ways. And potentially we can also study the breaking of symmetry in terms of the dimensionality, in terms of the directionality of the bonds by freezing a small group of bees, which was done before with fire ants by David Hughes. So that's another thing that we are hoping to do and get more information about that.
Good. I was also curious about in writing down the model, you have this sort of Leonard-Jones forces, and then you have a
Oh, yeah.
spring. And
Yeah,
I guess
so
intuition would have been you choose the Leonard-Jones, and the spring-like behavior is just what happens near equilibrium, but that's not what you did. So
yeah, so what's happening, we basically added the Leonard-Jones just to prevent too much overlap between bees, because we know that the bees in real life also have some excluded volume, so that's why we added it, and then we also have some simulations without it, which will work in the less dynamic case, where we just look at the stretching of the bonds. But once we move to, sorry, let me just move forward here to the agent-based simulations, and we want bees to start, moving somewhere along the swarm, then that's where it becomes important to define this excluded volume, so they don't just overlap with each other.
I think questions are starting to accumulate, but maybe we should let you get to the end of this segment, and then we'll go back over.
Okay, so I'm very close. I'm almost done. So I think just to quickly summarize that part, I showed you that honeybees are regulating the shape of the swarm, supposedly to become less susceptible to mechanical perturbations, and that they're sensitive to different things like frequency and forces and directionality. What we think is going on is that the bees are modulating the aspect ratio to minimize local deformations. And then I have a few things that I wanted to point out. The first is a little bit philosophical, and so take it please with a grain of salt, but I just want to highlight that at least according to that model and according to our experiments, the bees move from areas in the swarm that don't experience high deformations closer to the base, which is where the deformation is coming from. So potentially they're experiencing and enduring higher amount of mechanical forces for the greater good of the entire swarm. Another less philosophical aspect is that in a sense, the bees found ways to sample locally these large fields of elasticity and also manipulate them simultaneously to be able to maintain mechanical stability. And then lastly, I would just want to show you a little teaser for some of the X-ray experiments that we do in my lab today. So everything I showed you so far, we have no idea what's going on inside the swarm. It was all based just on the shape and what's happening at the surface of the swarm. And nowadays we're doing some experiments where we take the swarm into an X-ray machine, and we can do some 2D projections. We can also do some 3D reconstruction of the entire swarm. And we can get some really interesting information about, this is what I alluded to before, for example, the density profile in the swarm. So we can already see that the simple assumption we made about a uniform density along the swarm is not quite true. So there's a higher density closer to the base of the swarm, which makes a lot of sense if you think about the bees trying to minimize the average amount of load per bee. So it makes sense to have more bees at the top so that they can hold a larger amount of layers and support the swarm structure. And I think there's some really interesting analogies between what the bees are doing to other relations of strength and cross-sectional area, for example, in muscles. So we are kind of going in this direction, and we are calculating some of these power laws. It seems to deviate a little bit from what we know about muscles, for example, but we're trying to understand how the bees basically choose to modulate the density inside the swarm. So what you see here is the honeybee swarm version of that muscle cross section, but we have now a layer of a mass of bees, and that mass is going to hold all the bees that are underneath. And this is the power law that you're seeing here at the bottom right. So yeah, I think now would be a good time to stop and see if there's any questions about the mechanical communication.
So maybe I'll start with the things that are in the chat, and then other people can raise their hands. I think that if you raise your hand, you pop up in the list of participants, and so it's easy to call on people. So I'll ask you to do that. So two questions. One, do the outer bees replace those that are in the center of the swarm from time to time? I guess more generally,
yeah.
is the network relatively static, or are you seeing kind of?
Yeah, let me actually see. I might have a movie of that. So the answer is that it really depends on the temperature. Yeah, I do have that here. So ignore the X-ray, but I want you to focus on the movies here at the bottom. The movies here at the bottom, this is the same swarm, but at different temperatures. At high temperatures, there is a lot of motion, and therefore also a lot of exchange of bees from the surface of the swarm into the bulk of the swarm. And this hasn't been very quantitatively characterized so far, but this is another direction that we're interested in my lab, because having all this motion and exchange from the surface to the bulk poses more mechanical challenges that are at the moment not really clear how the bees are managing to have all this motion and still not break.
Okay. Another question from the chat is there a critical behavior with breakage due to vertical perturbation?
Critical behavior, in what sense? There is a critical force that would create perhaps start breaking a few bonds between bees, but then that crack would propagate, and then as I showed in the movie a few slides ago, this can lead to a larger crack that would basically get the entire swarm to break. I'm not sure if that answered the question though.
Well, if it didn't, maybe somebody will pop back up.
Okay.
Josh has his hand up, so you want to.
Hey, Orit, that was wonderful.
Hey.
I know we're only partway
Thank
through. So I'm curious to know more about the X-ray setup. What kind of resolutions do you have? How long can you run it for? What's the temporal resolution? Can you track well at that resolution, etc.?
Yeah, so it's not, I mean, it can always be better, but right now our resolution is high enough so that we can definitely see individual bees and reconstruct the bodies of individual bees. We can see even the two parts of the bodies, like the head and the abdomen. But that's it. So we cannot see the limbs, for example. It would be great if you could see the bees holding onto each other, but we cannot do it in this configuration. In terms of the temporal configuration, so it's an X-ray video. So basically, the video itself is recording at about 25 frames per second, which is great. But there's going to be a trade-off there between rotating the swarm at a certain speed so that we can do the 3D reconstruction. So if you're just interested in tracking individuals in a 2D projection, then I think this resolution is good enough. Did that answer your question, Josh?
Yes, thank you. Sorry, I had to find the
Okay.
Okay, we have one more question in the chat. From Diana, if you have a very viscous passive liquid and shook it in the same way, how different would it be? And so what's the signature of the system being active?
Okay, so if we have a viscous fluid that is being shaken, there might, if the water, if the liquid particles are the forces that they're experiencing actually get them to spread out, which I think this is, if I understand correctly the question, maybe that's where this idea is going for, then the swarm would become more elongated. If they're really being thrown out because of the shaking forces right and left, for example. More elongated in the sense I mean the base of the swarm. So it would we will see this breaking of symmetry so that it's no longer the base is no longer represented by a circle. It's going to be represented by an ellipse, for example. But that's not what's happening here. So I think that's potentially one difference. Perhaps there's more, but I'm not sure. Marianne has questions.
Yes, great. Yeah, so I was wondering sort of what's the threshold? Like, is there a like biological interpretation for like sort of this threshold above which the bees respond? Like, is it some high force or something to do with their mass or something?
Okay, so you're kind of asking. So in this plot here, I think we at least get a ballpark for that threshold. So if the acceleration is really low, then the shape of the swarm doesn't change very much. And then the jump, to the next level is from this yellow to the green line. So somewhere in between these two is probably the threshold, but we don't know exactly.
Okay, and that does it depend on the number of bees in some way, or it's just, I mean, you just measure like.
Yeah, so that's another great question, which so everything I showed you was for a fixed number of bees. That was about 8,000 bees. But of course, these forces are going to scale in some nonlinear way as a function of the size of the swarm. So I think it's a really interesting direction to take this to. What would be the effects of shaking, or even just flat on the shape of the swarm without any dynamical forces as a function of size, which is another thing we work on in my lab, but we don't. This is a bit preliminary at the moment.
Thanks.
Okay, maybe we should let you go on to the second chapter.
Okay, great. All right, so that was an example of communication, local communication within very large swarms of bees. In the example that I just showed you, the communication was more mechanical. And in the next example, I want to show you how honeybees solve a different kind of collective problems using chemical communication. Okay, so let me start by showing you this movie from an installation of a hive a couple of years ago in my lab. This is something that honeybee keepers do quite a lot, just for demonstration. So for those of you who are less familiar with beekeeping, that blue box that you see over here is the hive. So that's a commercial beehive. And it's really just a wooden box that has a very narrow entrance at one of the sides of the boxes. So that's what I'm highlighting now with my cursor. And then the queen is inside the box. So we place the queen inside the box. And then we place the rest of the worker bees a little bit further away from the entrance. And then we just follow what they do. So this is a very sped up. But you can see that the bees are sort of climbing, walking up the surface and find their way into the entrance of the swarm, of the hive. And if we zoom in to the entrance, we can see that there is a lot of bees that are fanning their wings. So if you had sound, you can probably hear all the buzzing. But if we zoom into one of these bees, they're not just fanning their wings, they're also positioning their abdomen in a very particular orientation. So what they do is stick their abdomen upwards, and by doing so, they expose a pheromone gland called the Nasonov gland. And this is this really thin white line over here that I'm highlighting with my cursor. That pheromone gland will produce pheromones, at a certain chemical concentration, and then the bees can sense these chemicals using their antennae. And they use this in order to potentially communicate with each other. What I want to show you is actually how they use this to find the queen and aggregate around the queen, which is a difficult problem because normally what the worker bees would do is sense the queen mostly based on her pheromone, so the way that she smells in a sense. But these pheromones decay very rapidly in time and space. So it's really not clear how the bees can find her from far away, which was what triggered this entire project. So we know that the bees use the chemical communication. We know that they expose this pheromone gland. And in a sense, this is really similar to another classical problem in biophysics, which is the aggregation of social amoeba. Again, by producing and creating and sensing the same chemical signals, these social amoebas are able to aggregate into spores that can later on help their survival under different difficult environmental conditions. So the bees do something kind of similar in that sense, but they do use one trick, which is airflow. So the bees expose the pheromone gland, and then they fan their wings, as shown here in this movie. And by fanning their wings, they drive air backwards along their body axis, and then they drive air on top of this pheromone gland, which gets these pheromones to diffuse in a very particular direction. And from some preliminary experiments, we noticed that when bees use this kind of communication when they're searching for the queen, worker bees that get close to the queen don't just walk all the way to her. They would actually stop at a certain distance and then start creating and amplifying these pheromones. This is a behavior that is called scenting in the honeybee research field. And then another bee would come from behind and would also start scenting and again until they propagate that information over large distances. So this is really just anecdotal. And then we wanted to study this more quantitatively. So Dumai, who is a graduate student in my lab, designed these behavioral experiments where we confine the bees into a semi-two-dimensional arena. So the bees can walk inside this arena, but they cannot fly. And the queen is going to be positioned in one of these corners of the arena, and the bees would be positioned at the other corner. And we just record everything from the top to get an aerial view. So this is how a typical experiment would look like. And you can see, for those of you with sharp eyes, you can probably see some bees fanning their wings. And if I scroll kind of faster through this experiment, and at the end, the bees do find a way to the queen and aggregate around her. But it's really hard to see, at least by eye or quantify by eye, how many bees we had performing this scenting behavior and in which directions. And that's actually really important if you want to understand the meaning and the physical meaning of these scenting events and whether they actually do lead the bees to the queen. Then we started collaborating with the lab of Greg Stephens, we created a high throughput classification based on machine learning of the behavior of the bees. In this particular case, we had these couple of hand labeled examples of bees that are scenting and bees that are not scenting. And the classifier resulted in, first of all, a yes/no question. So is the bee scenting or not? But also giving us the orientation, the body orientation of the bee. So in which direction this bee is scenting. So that was really useful because then we can use that method to get a high throughput analysis of some of our experiments. And here is the same experiment from the previous slide, now with overlaid with the analysis. So every time you see a blue arrow, that points backwards towards the direction in which the bee is dispersing the pheromones. So here's the same movie again. We can see that there is, first of all, a very sharp increase in the number of bees that are scenting in the beginning of the experiment. And then eventually that will decrease as the bees stop to aggregate. But what we really want to understand is there something special about the directions in which the bees are scenting that really helps them get the rest of the worker bees who are further away all the way to the queen. So to test that, let's think for a second about a hypothetical surface that maybe the bees are climbing up gradients in this surface. So then high values of the surface would correspond to the position of the queen, and the bees would just sort of move around the surface up the gradient. And to construct this surface based on the experimental data, what I'm going to do here is really take all of the scenting events and treat them as local gradients on this surface. So here I'm just showing you an example of this. These are the gradients, and then we're going to integrate them into a three-dimensional surface. So if we apply this on our experiment, this is again the same experiment that I've shown you before. On the right side here, I'm showing you the surface reconstruction. Yellow corresponds to high values of the surface. And you can sort of see that, I mean, it's of course fluctuating quite a lot, but in average, there's going to be some probability to get the highest point of the surface close to the position of the queen. Which we can do, we can actually calculate by just taking all of these surfaces and showing the average surface, which I'm showing you over here. So this is averaged over the duration of the experiment, and we can see that high positions along the surface are really going in good agreement with the position of the queen. I'm of course showing you just one example here, but we repeated these experiments several times. And that seems to be consistent. So it seems like yes, the answer is yes. The scenting events are somewhat associated with the ability of the bees to create a global map, in a sense, that would lead bees that are further away to the queen. But we wanted to understand better what could be triggering the bees to start scenting, and what is the decision-making process of this collective creation of the global map. So there's a couple of clues that we got from the experiments. The first one is that the decision to start scenting is somewhat probabilistic. So something that we can easily change and control for in our experiments is the number of bees that we have in our experimental arena, which is what I'm showing you here. So the different levels of green corresponds to different number of bees that we have in the arena. And we can see here on the y-axis number of scenting bees as a function of time. And we can see that the number of scenting bees for all of these different experiments with different number of bees in our experimental arena is proportional to the number of bees. So it's not like there is a very particular set of bees that are going to be the bees that are scenting, and is going to be a constant number of these bees. It's always going to be proportional to the number of bees that we have in our experimental arena. The second hint that we got from the experiments about what could be triggering scenting is the distance between bees that are scenting in our experiment. So we can take a snapshot of our experiments and calculate all the distances between the neighboring bees that are scenting. And we can see, besides one outlier, that most of the experiments, the distance between scenting bees is somewhat consistent, around 6 centimeters. So if really what's happening here is chemical communication, and a chemical communication that is following some kind of diffusion equation, then we know that a particular distance between the source of a chemical signal is going to be associated with a certain concentration of that signal. So perhaps what's happening is that the bees are being triggered by a concentration of pheromones above which they start scenting. So these are the 2 clues that we got from the experiments. And now we're going to take all of these ideas and information we got from the experiments and plug this into, again, an agent-based model to help us understand better this search and aggregation process. So the model is fairly simple. Like in the experiments, we have a 2-dimensional arena where agents can move around. And they are going to get triggered if they sense, if they enter a cell that has a concentration of pheromones that is above a certain threshold. And then probabilistically, in this case, 50% of the time, they're going to walk up the concentration of pheromones using a directed random walk. Or they're going to scent. So they're going to stop and amplify these pheromones in a particular direction that is associated, again, with the gradient of the local gradient of pheromones. And to do that, we really just implemented the classical solution to the diffusion equation, where we can have a parameter that controls the directionality of the signal, either isotropic, as shown here in the movie on the left, or directed, as shown here at the movie on the right. Okay, so when we run this simulation, we paid particular attention to 2 behavioral parameters. One is the threshold of concentration that triggers the scenting behavior. And the other is a directional bias that the bees create by fanning the wings. So 0 would correspond to having no directional bias, so it's an isotropic diffusion, and high values of this parameter would correspond to a high directional bias. And we can see a few interesting regimes that are actually perhaps not very surprising, because they occur in many aggregate systems, not necessarily active ones, of aggregation of materials. So let me just go through some of these regimes. At very high concentration values and high directional bias, of course, nothing is going to happen, because the concentration threshold is so high that none of the bees is going to get triggered. So there's no clustering. At very small values of these parameters, we're getting into problems of local equilibrium, where the bees create small clusters that are really hard to break and then aggregate around the queen. At intermediate values that are happening closer to the regime of no directional bias, we see something that is similar to diffusion limited aggregation. So the bees would basically perform a random walk, and if they get closer to the big cluster that is forming around the queen, they would attach to it. And of course, this process of aggregation would scale very badly with system size. And then there's this sweet spot that I'm showing you here, highlighted in red, that really opens up when there's a non-zero directional bias, and that's clustering via percolation. And these kind of simulations that I'm now playing here at the bottom right, there's going to be these chains of bees that get triggered, and they trigger each other to scent, and they propagate throughout the entire system, and then also aggregate into one large cluster around the queen, which is of course the optimal configuration. Okay, so I think some takeaways, some quick takeaways from that part. I showed you that the local positions and directions of descending events do integrate into something that we can think of as a global map that leads the further away bees towards the queen. And the model that we created was helpful in a sense of understanding why this this directional bias could be beneficial for that system. And we showed that in having this directional bias can actually help the bees avoid configurations that are going to result in local equilibrium, like the small clusters that I showed you before. Okay, so I think that's all I had to say about this problem, and maybe this would be a good time to stop and see if there's any questions about the chemical communication or bees in general before I move to talk about fireflies.
So we have one question in the chat, and then maybe other people will raise their hand. The question is the swarming behavior purely chemical in nature? In particular, could there also be wing vibrations that are producing some sort of song or physical signal, like we know happens in Drosophila mating?
So we know that this chemical communication is at least important and plays a role in the aggregation. And bees use it on several different occasions. For example, I think the first quantitative characterization of this process was of bees stand at the entrance of the hive, and they produce these pheromones. And in hives where bees were actually sending outside, foragers who come back from longer distances are more likely to find the way back into the hive. And then so we know that it plays some role. And then, of course, an excellent question is there any other sources of information that the bees could be using? And well, we cannot rule it out. I would never want to rule out any other sources of information because we know that the bees can also be using mechanical communication, as I shown in the previous examples, but also maybe more famously using the waggle dance. So we haven't really focused on this kind of communication. And I think what I can say, in confidence is that the chemical communication does play an important role, and it can explain the behavior of the bees, but I wouldn't completely exclude other kinds of communications.
Other questions? People want to raise their hand or in chat? Well, we can also come back. So maybe maybe you should move ahead and tell us about fireflies and
okay.
Take more questions.
Yeah, sounds good. Okay, sounds good. All right, so I'm gonna switch gears and move from honeybees to fireflies, and from mechanical and chemical communication to more visual communication. So fireflies communicate using the lantern in their abdomen. They usually do this more often during their mating behavior. So the males would flash at a particular pattern, and then the female would respond in a particular flash pattern. And this the signal it's a visual signal. It's approximately digital, so light on and light off. And it's potentially traceable in very large groups, as I will show. And I think that it's a really nice system to use when thinking about some of the aspects of communication that I mentioned in one of the earlier slides. For example, signal-to-noise ratio and compression, minimization of information, because of the simplicity of the signal. And of course, fireflies would need to deal with these kind of communication problems because these mating swarms are very large. So they can be thousands and thousands of individuals in each one of these mating swarms. And they seem to be using two kinds of strategies to minimize visual clutter. One is, as I'm showing here in the movie on the left, is to synchronize their flashes. So of course that if all the communication channels are producing the same communication pattern in a synchronous way, then that immediately helps increase the signal-to-noise ratio. There's other really interesting class of fireflies who don't synchronize their flashes. And another project in my lab that I won't have time to talk about today is how some of these fireflies might be designing their flash pattern in a way that is going to make them more detectable at the expenses of flashes coming from other species. But for today, I think I'm just going to focus on the synchronous fireflies. And I'm sure all of you are aware that synchronization is a very common phenomenon in nature. It doesn't only happen in living systems. It, of course, also happens in inanimate objects, for example, electrons in a superconductor, or larger objects like these pendulum clocks. But they do also happen in living systems. For example, us humans tend to synchronize our behavior, for example, in some parts of the world, not so much in the USA, but at least in Israel and in Europe, people tend to synchronize their claps at the end of a concert or other shows. And we also know that musicians, of course, synchronize their behavior to produce beautiful music. And then fireflies are actually another landmark example of communication in nature that is synchronous. And it's often mentioned in textbooks, for example. And some of the main models that account for this kind of synchronization processes assume that each individual in the group of agents or in the group of people or in the group of electrons perform some kind of periodic behavior that can be represented by a period length and some phase within that period is a function of time, very broadly speaking. And then one way to introduce synchronization between a group of such agents is to define coupling between the phases or differences of phases in pairs of these agents. One very famous model to do that is the Kuromoto model. And there's many ways to implement it. So in here, I'm showing you an example where there is a coupling constant K that couples the difference in phases of pairs of agents in that system. K can be a constant. It can be a function of the distance between the individuals. It can be, of course, more complicated function of that distance and more and more and more. I'm not going to get into details here, but I'm just showing this slide to show that there are so many variations of this model that can result in very different kind of behavior and onset of synchronization, but not a lot of quantitative data with fireflies, at least, to test whether and how well these models work. So that's the gap that me and my lab members wanted to fill, in a sense. And this is a project that was led by Rafael Safati, who is a postdoc in my lab, and Julie, who is now doing her PhD in New Mexico University. And we worked with a species of fireflies called Photinus carolinus. They are notoriously hard to grow in lab conditions. They're very finicky, and I can talk more about why after if you ask me. So our best bet was really to just go straight to the source, where we know we can find them in nature, in their natural habitats, and that specifically for these species, they're very abundant at the Smoky Mountains, specifically in the Tennessee side, which is where we all drove and performed some of these experiments. And these fireflies produce a flash pattern that has bursts. So this is an illustration here of the time series of the bursts of a firefly swarm. And it really looks like it is going to be a flash on, flash off several times. And then there's a quiet part, and then again a burst. And they continue this pattern on and on for a few hours every night, but only for about 10 days out of the year. So you really have to be there at the right time to see it in person. But if you can't, then hopefully you'll appreciate this movie. This is one of the recordings that we made with our cameras. So what you just saw was one burst, and now is the quiet part. And here is starting a second burst, and on and on and on. Okay, so there's actually quite a lot of useful information we can get just from that one view of a camera. So it's a temporal information in that sense. For example, if we plot the number of flashes on the y-axis as a function of time, and we do so for low firefly density, so there's not a lot of flashes here earlier in the night, at least, and later in the night. And compare this to a situation where we have high firefly density, where we see this burst structure emerging. So we can quantify that transition in different way. One way that we chose to do this recently was to look at the variance of the number of flashes plots, and I'm showing here on the left side. So of course, for low firefly density, since there isn't a very particular structure there, it's going to experience a different power law, something that is maybe closer to a Poisson distribution, in comparison to the power law that we would get of the variance as a function of the number of flashes when we do see these synchronous patterns of bursts. Okay, but I did show you before that some of the mathematical models that we wanted to put to the test account for distances between the individual fireflies. So a 3D reconstruction would be really important here. And that's where some method development went into place. We had one setup where we had two cameras that are placed kind of outside the swarm, and we're just recording the swarm as a whole. This is just a very classic setup for a stereo vision. So all the math was there and it was really easy to use. We also had a different version, slight tweak of that, where instead of having external cameras, we had two cameras that has 360 field of view, and we placed them right in the middle of the swarm. And so in a sense, we're kind of getting the point of view of an individual firefly from that swarm. And here there was a little bit of new math development, and which we published last year, but it works. So we can get the 3D reconstruction using both methods. And I also just want to mention that this new method with the 360 cameras, we use relatively affordable cameras. They're really just GoPros off the shelf. And because they're so easy to use and potentially suitable for citizen science, we partnered with National Geographic to create a platform where people, hopefully at some point in the near future, would be able to go by natural environment with potentially some fireflies and turn this into a citizen science project where we can acquire data about potentially many different species, potentially in many parts of the world. But I think the most important point is that we can do this 3D reconstruction, and here is an example of that reconstruction of the flashes within. This is with the 360 cameras setup, so we get data about 10 days for a particular swarm and a few hours every night. And we can start getting some really interesting statistics about the spatiotemporal relation between the distances between flashes and the time between flashes. So this is what I'm showing you here. On the y-axis, we have the distance between each pairs of flashes that we recorded in our setup. And the x-axis is the time between these flashes. And in this particular example, we're seeing two trends. There is this blobs of high probability that are up here at around the same distance, but at different times. And the time is correspond to the time between flashes within a burst. So these top blobs over here are corresponding to fireflies that different fireflies that flash within the swarm. And then these bottom ones here, the red blob here that then move a little bit upwards towards higher distances and so on. These are more likely, because the distances are lower, potentially coming from individual fireflies that are moving, and along their trajectories, of course, there's going to be some correlations as well. Okay, so that's one piece of information we can get from these experiments, and we can think about ways to incorporate them with some of the mathematical models that I mentioned before. But that's actually not the end of the story. So there's a few other interesting observations that we made that kind of made us think that we need to rethink some of these mathematical models and perhaps come up with modifications that would allow us to better agreement with their experimental observations. And okay, so let me see. So one interesting observation was that there's not only spatial correlations on a small scale that I've shown you in the previous slide. There's also spatial correlations and propagation of information that occurs on larger scale. And in particular, that's within the onset of flashes within a burst. So let me try to go through these plots. What I'm highlighting now with my cursor is really just, again, the time series of flashes is a function of time. And if we think about the phase of an individual as the time that it started flashing within the burst, then we can identify early flashes within a burst and assign them a low value of the phase and vice versa for as it progressed throughout the burst. And we can see that on average now over here, what I'm highlighting with my cursor is an aerial view of a section of about 30 meters long in our recording. And we can see that in this particular case, the flashes started closer to the camera here on the left side, and they propagated away from the camera to the right side. And I'm also showing here a movie that captures this behavior. What's important to note is that there's a separation of timescales here. So we know that the speed of a firefly is about 0.3 meters per second, but the speed of this front of propagation of the wave that I showed you is an order of magnitude higher. So it cannot be explained by the movement of the fireflies, and it has to be explained by some kind of interactions, local interaction between the fireflies. And then perhaps the more significant observation that we made was about regarding what individual fireflies do. What I showed you so far was outside in their natural habitat. And I forgot to mention that we're really just seeing a small fraction of the swarm. So the swarm spans miles over miles, and we actually never saw the end of the swarm. So we can really just get a recording of a small part of the swarm, which means that we don't really know what was the field of view of individuals and what other individuals outside of our recording they may have interacted with, which is why we wanted to have some more controlled experiments still in the natural habitat, but we brought with us to the field these black tents that are opaque to light. So we bring a bunch of fireflies into these tents, and then we know exactly how many fireflies we have, and potentially also we know that there is less occlusion from vegetation and so on. So we can perform these experiments and we can track individual fireflies. One major limitation of the method is, of course, that we can only see the fireflies when they flash. So here we are interpolating between flashes to reconstruct the trajectories, approximated trajectories of individuals. But we can see some really interesting trends in terms of their ability to synchronize with each other. So if we go back to just a reminder again of the pattern of the flashes, there's two important time scales here. One is the flashes, the duration of flashes within the burst, so that they will have a certain period, and then the flashes between consecutive bursts. So there's going to be the time scale between the onset of one burst and the other. And if you look at the shorter time scale within the burst, we can see that fireflies can actually synchronize fairly well with each other, even in very small groups. So what I'm showing you here is a tracer plot of flashes coming from 3 different individuals. And you can see that within the burst, they're doing a fairly good job aligning their flashes with the other 2 individuals that are present in the tent. There's some other interesting behaviors, for example, the relay of the signal. So here, for example, at the top plot, the firefly is represented by the color blue, is starting the flash, and then others join him and carry on longer than that original flasher. But the point is that they're doing a fairly good job aligning their flashes within a burst. However, if you look at what happens between bursts, and we look at that time frame, something sort of unexpected showed up, which is that at high number of fireflies, we get a very characteristic interburst interval. But as we reduce the number of fireflies in the system, that interval becomes less predictable. And here are some distributions of that property. So on the y-axis, this is a probability distribution function of the interburst interval. At the bottom plot, these are 20 fireflies, and they have a pretty consistent peak around 10 seconds, which is what's happening outside. But then if we reduce the number of fireflies to 5, then this distribution becomes wider and even more wide for one firefly. So of course, these observations are in disagreement for some of the Kuromoto model assumptions, which is that individual agents or animals would still perform this periodic function even when they are taking away from the swarm. So that it doesn't seem to be the case, and we are currently working on developing some extension of these Kuromoto models and other sort of integrating fire models to account for that these observations. Okay, the last thing I wanted to show about fireflies is again just a teaser, but I think it can be a really interesting direction to take this to. So it turns out the fireflies do respond to artificial light, and we can do some entrainment experiments with them. So as the fireflies by themselves, they're not very well represented by oscillators, but we can sometimes entrain them with artificial lights. So it's a really simple setup with just an LED that we can easily take with us to to the field, run by Owen Martin, who is a new graduate student in my lab. And it seems to be giving some interesting preliminary results. What I'm showing you here is a couple of time traces. The top one is the LED, and the bottom one is the firefly. And you can see that the firefly changes its frequency as a function of the frequency of the LED, and we can run this over several different frequencies of the LED and calculate the response of the firefly. And you can see that around the natural frequency of the firefly, which is around 700 milliseconds, we see a very different behavior as we deviate from that natural frequency. So again, really just a teaser. I can elaborate a bit if you ask me later on. Okay, so I showed you that we can finally record some firefly flashes, both in their natural habitats, but also in some more controlled environments. We talked about the temporal onset of synchronization and some long-range spatial correlations. And some of the aspects of small groups of fireflies and individual fireflies who don't act as oscillators when they're by themselves, and how we're going to use these observations to come up with new mathematical models is something to be shown later. But if you also have some ideas, then I would love to know. And there's one last thing that I want to just quickly highlight. It's really more for those of you who are interested in history of science, but I basically showed a couple of examples where animals in large groups, in particular insects, communicate with each other using different kinds of communication, mechanical, chemical, and visual. But since I'm also in the computer science department, we also think a lot about some applications of these observations we made of the natural world. And there was a really nice article that came out just this year, a few months ago, about an interesting interaction between Albert Einstein and von Frisch. I'm sure you all know Albert Einstein. For those of you who don't know von Frisch, he's a Nobel laureate who discovered, for discovering the communication language of honeybees of the Weigel dance. And it's a fascinating story. It's based on a lost letter from 1949 that Einstein wrote right after meeting von Frisch in Princeton University, not far from where you are. And in this letter, there was some kind of discussion about potential applications of von Frisch observations of honeybee communication to potential technological applications like new radars and so on. And of course, Einstein was right. And I just wanted to highlight that there's a lot of interest in these kind of local interactions in large groups of individuals, specifically for technological applications in the field of swarm robotics, where there's many individual robots that are potentially replaceable, but they can perform some interesting global function. So I think that with that, I will stop and just thank everybody in my lab. This has been a collaborative effort, postdocs, graduate students, beekeepers, undergrads, and even a few very talented high school students that joined us, collaborators, funding, and thank you for your attention. And I'll be happy to take questions on the fireflies, but also any parts of the talk.
Great, that was great. Thank you.
Thanks.
Well, we have a couple of questions in the chat, so let's start there, and then people can raise their hands. It's a pleasure to have Gordon Berman join us from Emory University. Gordon's been involved in problems surrounding the physics of behavior, I think, since the beginning of resurgence of interest in this, and had the pleasure of working together for some period. Since he's been at Emory, he's branched out into much wider range of problems. And again, in addition to the science that you're going to hear about, he's been very active in education. Obviously, we're professors, so you have some day-to-day responsibility. But outside that, he's been very involved in summer schools around the world, bringing this perspective of the sort of physicist approach to the quantitative analysis of animal behavior to much broader audiences, both on the physics side and the biology side. And I think many of those schools have been quite influential in the growth of the field. So, Gordon.
Thanks, Bill, and thanks everybody for the invitation and for the opportunity to speak. So I do have my chat open here on my screen. I'm able to see it. So just whenever you have any questions, feel free to either interrupt and mute yourself, or just type something into the chat, and I'm happy just to sort of go sort of try and answer things more or less as we go. So this is sort of my cute first slide that I wrote for this, because really thinking about themes and variations in animal behavior, because what I'm going to try and have this theme sort of running throughout the talk today will be thinking about how do you really understand variability that we see in behavior, behavior being in some sense the thing that has a huge amount of variance in it that most people throughout science that are trying to quantify things tend to sort of throw up their hands in the air and say, I can't do anything with it. So, but it's really focusing on that variability. But so this is kind of the pretty picture of what I'm trying to see, but how I often sort of feel when I'm working in this field, and with apologies to Raymond Carver and Edward Hopper and Banksy all at the same time. Oops, why isn't that? Oh, there it is. This is sort of what we tend to sort of think about, what we talk about when we talk about variability in behavior. And other than the fact that I just absolutely, just every time I see this painting by Banksy, just makes me laugh a little bit. It kind of has that picture of, in a lot of sense, what the problem is when we're really trying to think about and trying to develop quantitative theories of what behavior is that there's so much capability and there's so much subtlety in any of the interactions that we see across these different individuals in the scene, and even as a sort of the scene as it is. So we're going to sort of move through sort of notions of variability, how we kind of think about it in our group, and how we're trying to move forward. So maybe just step back a second. Like what we do in our group and what we think about is really trying to understand natural, or in some cases naturalistic, behavior. So for example, anything from watching this fly move around in the lab, which is sort of one of those data sets where we just keep on getting more and more interesting stuff the more we stare at it. So other things like the collective behavior that Orit beautifully showed in the first part of today's workshop, to other sorts of animals like prairie voles trying to desperately reach their partners, or animals running through more complex environments. And a lot of what this is, and what we try and do a lot of the times, how do we start to quantify and put numbers, quote unquote, on these sort of types of systems. And the types of numbers we've gotten pretty good at in the last few years is all of these types of automated tracking techniques for extracting things from images, like deep lab cut and leap and others, with an ID tracker and all sorts of more kind of artisanal methods that people have had using deep learning techniques to take what would take hours and hours or days or weeks or months of time when I was a grad student, and really sort of speed up this process of data collection. But this is just sort of what's happening from moment to moment, or just over the course of less than a second dynamics that we're seeing in the animal. But the sort of things we tend to really be interested in our group are these longer time scale structures in behavior. At the end of the day, behavior is really almost this argument between all of these internal states which are fighting with each other, and are coalescing to bias the types of actions an animal makes. The example I like giving is if you're lying in bed sleeping at night or hungry, you decide whether you decide to get up and get food or you decide to stay in bed. So you have these sort of internal states of hunger in your circadian cycle, which are kind of fighting with each other. And on top of that, there's the other states that are like social affiliativeness or aging or other types of things that we also think about. And how do those things bias the actions, and how can we build models across these multiple scales? Fortunately for us, there's been a whole line of thinking in classical ethology, starting from basically over the past 60 to 70 years. So Orit showed von Frisch. I'll show Tinbergen here, who is this co-Nobel laureate, thinking about this notion of hierarchical structure and behavior, and how behavior really can be thought of as having these sort of, again, interacting states, or even other sorts of work where we look at more dynamical processes where we have sort of low-level dynamical systems which are generating more low-level features, or even we have things like Richard Dawkins, who actually was a student of Tinbergen's, and before he was the Richard Dawkins we all know, and was actually a fly grooming expert, he came up with a bunch of sort of thinking about how does an animal organize patterns and sequences. So a lot of, only half joking, but I say a lot of what my group's work is really thinking about how do we take sort of philosophical or and linguistic pictures like this, and how do we turn them into mathematical theories and models and predictive notions that we can use to really gain insight into systems. And so that leads us to sort of the first bit of the talk, which is about measuring behavioral variability. So how do you go, like, even before we try and make sense of this variability that we see in the data and try and understand it and predict it and understand the underlying causes, how do we even decide to measure it? How traditionally, like the most common way of doing this is the standard way. So you would have like differences in mean postural trajectory across trials. Right? You have some trial average where you put some animal in some apparatus where it reaches or it does something along those lines, where it does an isocod, and you measure sort of difference at the time series, you get some variability, you calculate the mean, and then you actually measure that variance. And then there's been a bunch, and along these lines, there's been a bunch of beautiful work that's come out of this dynamical systems and neuroscience notion in the last few years, really trying to think about how do we extract and how do we encode these sorts of things in these types of environments. But this gets much harder if we don't have trial structure. Because in real normal ethological behavior where an animal is just running around in the wilderness, sure, there's sort of discrete stuff it does, but those discrete things it does usually within a larger sort of overall framework. So what we need to be able to do is have a way of measuring behavior that allows us not only to be able to move from all these different types of behavior, but also that allows us to be able to coarse grain out from these different time scales, because behavior is something which doesn't exist at a single time scale. It could be a twitch of a muscle, it could be reaching an arm, or it could be longer time scale things like foraging, or even the simple act of not falling asleep. Right? So these are kind of, which maybe I'm failing all of you at right now, but the basic idea is that how do we actually build something from that? And so this first part, I'm going to try and zoom by on this part, but this is all rather old, and I'd rather try and get to some of the newer stuff now. But sort of the notion that we've built, and what this is work in collaboration with Bill and Josh many years ago right now at this point, is thinking about how do we like isolate the stereotype behaviors an animal performs in a data set. So for example, if you imagine the state of all possible postures. So could be like the joint angles of my arm or something. I could move my arm like this. You can tell I studied insect flight for my PhD. You could have some more periodic motions like this. You could have some crazier things, or it could fill up the whole space. But if you actually watch an animal behave, what we kind of make the bet on is that what we don't see is this, but what we see is something which looks more like this, where you see a bunch of behaviors that are performed maybe with slightly differently each time with a little bit of variability within them. But a lot of the sort of variance in the overall kind of postural trajectories of the animal are explained by, there's a sets of stuff that they do a lot. And every once in a while there'd be some weird things, but overall there's some sets of stuff. So we'll call these our stereotype behaviors, these kind of postural trajectories that an animal performs often and repeatably, and then we can try and isolate those. And then what we can do to try and look at the longer time scale dynamics is we can see how it kind of moves between all of these different types of stereotype behaviors. So we've developed some techniques to do this. So this is way back from this 2014 paper at this point, and there's code to do all of this thing. But really what we're doing, and sort of the key kind of feature here, is what we take time series. And so we take those time series, like, for example, the track joint angles of a limb, or basically the opening angle of a wing in a fly, or the head angle of a rat. And then we basically look at the spectral properties of those time series. We see essentially what parts of the body are moving at what speeds. And then only from there, then we can do whatever sort of embedding procedure we want. Here we use t-distributed stochastic neighbor embedding. You could do other things, or not even embed if you wanted to, but really the key is that we take this high dimensional space and of postural motions. We look at the dynamical features in those data using a wavelet transform, which is sort of this multi-time scale short thing that's like a short time Fourier transform for those who haven't seen it before. And then we try and embed it in this space where points that are nearby are represent the animal moving similar parts of its body at similar speeds. We do that, we get this space. So this is looking at a bunch of data from flies, Drosophila melanogaster, running around within sort of this featureless environment for long periods of time. And so, for example, we can pull points in time where we sort of map the fly to that bottom right part over here. And for example, all those points there, the flies are running similar speeds. Or up here, the flies are grooming their head. Or over here, the flies are grooming their left wing. So as a result, we sort of call this thing our behavioral space or behavioral map, depending on what we're talking about. And so where we basically, each of these peaks correspond to different stereotype behaviors, and we can sort of watch to see how it moves and hops between these different peaks. One of the things which is going to show up a lot, and so we'll get back to this more, is that perhaps unsurprisingly, because we designed this space such that points which are similar parts of the body are moving at similar speeds or grouped next to each other, we can sort of, if we look at the videos coming out of those things, we can put sort of names on them, things that kind of look similar to us, and different parts of the space sort of have similar names. So over here is idle, is slow. This is actually proboscis extension up here, anterior movements, posterior locomotion, and other kind of more subtle things in the middle. So we can build this type of model, and then the point is this is the starting point. So the goal is to think about how can we use this type of a model where we can imagine sort of making these maps or seeing sort of what part of the map do different animals or different types of age groups all fill up within the map, and how can we then see how that's changing either over time or across individuals, and try and understand some of the data from there. So this is a couple of examples of the first way, which is, let's just see sort of how the repertoire is changing. And so these are some results from data that was actually taken in Josh Davids's group that we recently put on archive. This is the work of Catherine Overman, who's a grad student in my group, looking to see how these behavioral maps are changing as flies aging. We have kind of a cool story here, thinking about basically where a lot of the changes in age seem to be highly correlated with changes in energy metabolism, which might point to, which is sort of is in agreement with a lot of thoughts that people have about aging and metabolism and how that affects behavior. If you're curious more about that, just take a look at the archive paper. But what I want to spend a minute more on here, really thinking about how you can analyze how repertoires are changing, is to think about this, because this is going to come up a few more times over the course of the talk, where this was a data set taken by Jessica Candy, who is a postdoc with David Stern, who's been a collaborator with me for quite some time now, and as well with from some little fly lines that were developed by Hiro Miki, working with Gwyneth Carr and Wyatt Korf. And really what we were trying to do is look to see what the behavioral effects are of stimulating a bunch of neurons in the fly's ventral nerve cord. So for those of you not looking at fly brain, this is the fly brain. There's the head and then the ventral nerve cord. And what we were curious about is because in the fly head, you have, say, order 10,000 neurons, which then goes down to order about 600 neurons in the ventral nerve cord. And it's through those 600 neurons that almost all behaviors are controlled. And so could we try and tear apart sort of the connections between brain and behavior to see how this compression winds up occurring? And so then we can say, because they're flies and because there's beautiful fly magic, you can make a bunch of fly lines where you can express channel rhodopsin. So a red sensitive channel, ion channel, which opens when you shine red light on it, in only of these pairs of neurons, so these sort of symmetrically paired neurons that you shine in the fly brain. And then basically ask, if you shine a red LED in the background, because the fly's cuticle is translucent to red light, what happens behaviorally? Can we make sense of this? And we can do this across all these different lines because of sort of all the fly magic that our great collaborators have been able to use. Oops. So Jessica made this setup where basically we have this, where we have this red light, which is turning on and off. So it's on for 15 seconds, off for 45. And then we basically try and isolate what are the parts of the map which are getting quote unquote excited as we excite the neurons with red light by activating the neurons opportunistically. And if we do that, so here's one example. So here's a sort of it looks like about 6 pairs on each side. And essentially what you'll see is this is a bunch of trials averaged together. And so at t equals 0 seconds, oops, we'll see that the light turns on. So, and basically all the behavior winds up coming to this sort of region of this behavioral map. Note how this is a different sort of overall behavioral map because of different data. And at 15 seconds, it turns off. And so you basically get this activity that sort of goes back and sort of spreads out sort of overall thing. You can ask what part of the space got sort of quote unquote activated and inactivated. And so you can see you get a negative region here, which turns out this is idle. And so the animal is not doing nothing when you turn on the light. And this region is positive, and you, as you sort of saw, you saw all that sort of density kind of pop up to the top. And you can see that this happened reliably trial after trial after trial. You can ask what the fly is actually doing. And to the left, light turns on, and they're grooming their head uncontrollably. And we can do this. That was just for one line. That's like this one up here. We can do this for a bunch of different lines, each sort of covering with a statistically kind of upregulating region of a different part of the space. And we can use these types of methods to figure out what stereotype behaviors are changing and really trying to link kind of this variance and neural behavior to behavior, neural dynamics and behavioral dynamics. And we'll come back more to this later. But so the type of variance that I'm particularly interested in, what my group is more or less focused on more recently, is thinking about variability in behavior across time. We can start back up with this behavioral map over here. Again, with each of these peaks corresponding to different stereotype behaviors. Then we can ask sort of how on average does this animal move through the space? And we have a bunch of peaks and we have a bunch of transitions, so we can, we're physicists here, so we can go and make a Markov model out of it. Basically, we can say, all right, here's one point here, and the curvature is the direction of transmission. So for example, this is here because it's right-handed. And then the thickness of the line is the Markov transition probability. So given I'm at this state, what's the next state that I traverse to? We have this overall kind of structure. And the first thing we see is this isn't a hairball. Right? We see that there aren't that many connections that are going kind of from here to here. Instead, we get most of these sort of local connections to the space where the animal is transitioning to nearby behaviors. Might ask what the reason for that is. Some of that reason might just be for a fairly mundane reason, which is postural facilitation. Notably that if I start grooming my head, if I'm a fly, I need to go and rearrange a bunch of my legs around before I start grooming my wings, otherwise I'm gonna fall flat on my face. So I can't go straight from here to here, I've got to go through some intermediary steps first. Similarly, I can't go from 0 to my fastest speed. So I can't go from here to running as fast as I can without going through a bunch of sort of intermediate gear shifts. So this is sort of slow to fast along locomotion. But this is this Markov picture. And this is kind of the picture that most people have tended to use when describing how behavior transitions, including some great people that are part of the center. Thinking about kind of these, actually, and all of these papers I like very much, but what you're essentially doing is you're going from one behavior without taking into account too much history in the past, maybe order of a second or so, and trying to predict what the next thing the animal is doing is. But if we saw kind of, and thinking back to kind of these more ethological pictures, right? If we look up here at Tim Bergen's sort of famous hierarchy diagram, like building is like, this seems like a time scale that would be more than just a second, right? You're going to go to build a nest or to construct some or to other sorts of things. This would take far longer than a second, or maybe far longer than minutes or hours or even days, right? In order to have sort of the state. So the notion that you don't have any history dependence, this feels like something that we should probably test. And so this is what our sort of our next step was. And so this was these were some results that in a paper that I wrote with Bill and Josh back in 2016. And what you can basically see is we can ask this question: do behavioral transitions remember? And to a certain extent, we can also ask, how much do they remember? We have these sets of transitions. So what we can do is, first of all, this is essentially a transition matrix plotted in a really pretty way. What we can do is let's actually go back and call it matrix again. And these are the exact same data here. Just now, instead of having all those lines in the graph, I'm actually plotting the transition matrix, where this is the initial state of the flies, this is the final state. And I've sorted them sort of based on these coarse-grained regions like idle, slow, anterior, posterior, and locomotion. And what you can see, first of all, is this looks pretty modular, right? You have, and that kind of agrees with that intuition we saw, that we're mainly having these sort of localized transition structures in there. But we can actually use Markov theory to ask sort of how much do these things remember. If you really take a Markov model, what you're doing is you're taking that matrix and just multiplying it by itself 100 times. And if you do that, what you get is this matrix here. So this is just assume looking 100 steps in the future. And what you get is a bunch of vertical lines. And that basically means you're transitioning to the mean distribution, because it's more or less independent of my initial state, or which state I go to, the probability of it is more or less independent of my initial state. We can actually measure the joint probability distribution from the data. And if we do that, we actually get something which has a lot more structure to it. And in fact, it still maintains a lot of that modular structure that we just saw in the previous slide. And even if we go at 1,000 time steps, so an order of magnitude, we still can see kind of the remnants of that structure there, which are way more than what we would have expected in the Markov model. And so we can quantify that, that sort of visual perception that we have by looking at the eigenvalues of that transition matrix. So by the Perron-Frobenius theorem, what we know is we have, because these are all transition matrices and they have to sum to 1 along each of the columns, here each of the rows, essentially we know that we have one eigenvalue which is equal to 1 OA, so we're going to ignore that. And that's basically corresponding to the eigenvector, which is our steady state distribution. And then we have a set of lower eigenvalues, which all have to have a absolute value less than 1, which should decay off. And in fact, for a Markov model, they're just going to decay off exponentially because we're just multiplying that matrix by itself over and over again. So that's sort of these dashed lines here. They're not quite exponential because there's finite data and other types of effects that we have in any real experimental system. But if we actually look at what we're getting from our data, we get these solid lines here. And note how this is the second eigenvalue, third eigenvalue, fourth, fifth, sixth. And note this is a log plot. So we're getting that these sort of models are 2 orders of magnitude bad. It's not just sort of a little bad that we need to fix something in the second decimal place. This is sort of, there's a fundamental problem in trying to understand behavioral sequences this way. So what we try and do then is, what we've been thinking about a lot lately is how do we actually write down models that can do that. Now, one thing to point out is that we could ask, how generic is this? Is this just a weird thing about fruit flies? So we can write this same thing for, well, still fruit flies, but now fruit flies all a bunch of different ages. So this is also appearing from Catherine's recent archive paper. And also looking at rat behavior. So these are some data taken by Jesse Marshall, who works in Vincent Ovesky's lab at Harvard. And basically looking at, or we get the same sorts of things from rat behavior as well, these types of time scales and time series, or even, so this is work from an undergrad and senior thesis that Yatang Yang did a few years ago now. We could even see these same type of structures in human brain ECOG data in epilepsy patients. We play the same sort of game of time series coming from ECOG data rather than from behavioral data. So this is a pretty generic phenomenon, but yet if you actually try and write down models that do this, that are of the sort of standard statistically stationary models, they don't tend to work very well, and you can't tend to get these types of results. In particular, it's very challenging to get multiple eigenvalues, which all have this kind of high non-Markovian structure. You can always sort of engineer a relatively simple system that can generate one or maybe two eigenvalues, which have this relatively slow decay. And you can clearly get this if you were to write down some sets of epicycles in a hierarchical hidden Markov model that could generate these things. But none of the standard sort of relatively simple, straightforward, not infinite number of parameter models tend to generate this stuff. So what we were then curious about is how could we then actually isolate what are the features of this? And one question you could ask is, what's the mutual information between sort of the behavior I am now and then the behavior that the animal is performing sometime in the future? And if you do that, what you can do that calculation, a Markov model is in red and the data is in blue, and as perhaps unsurprising based on the previous plot, you can see that the data is lying above the Markov model. And the error bars here are smaller than the width of the line, just because it's based on so much data. But then we can ask, all right, let's take a look, say over here, right? There's not a huge difference in bits, maybe a couple of tenths of a bit. Here really is the difference. But we can ask, of all of the transitions, right? Because this information is basically looking at sets of transitions between, or sets of pairs of behaviors rather, and basically looking at a various time point, what are the contributions to this overall information? And you can ask, what are the pairs of information which are contributing more to the information than you would naively expect in a Markov model at that time? And if you do that, you all get these within region pairs. You basically all get these sort of within region pairs of behaviors, which suggest that, and you can actually measure this from the data, that you're sort of that the regions tend to be kind of sticky. That you get in one of these regions and you stay there maybe longer than you would naively expect you would based off of a Markov model. And so we then asked, okay, again, we saw this, can we confirm this observation by then trying to cluster the space and see whether in fact that is the best way to represent things. So this is where we use an approach which is familiar to many people in this audience, thinking about it in terms of an information bottleneck approach, where we're going to try and cluster this behavioral space. So from our states S of t to some partitioning Z of t. And we're going to try and do it, so we're going to optimally predict the future. So increase the mutual info, have as large of a mutual information between the partitioning and the future stated system, but having as simple of a clustering as possible. And then we can vary, and we have our Lagrange multiplier term beta to basically vary the relative importance of those terms. And then we can also look at different numbers of clusters of things. And if we perform this optimization, so where we have this simplicity on the x-axis and the predictability on the y-axis, what we can see is we get these sets of curves, where these curves are kind of decreasing comfortably the further out in time we look. So the further out in time, the less we know about the future. This is a good double check that we haven't done something completely stupid. And then what we can do is we can move along one of these curves, say tau equals 100. And if we do that, moving kind of up along one of these curves, moving from a simple description to a more complex description, what we see is the space has this hierarchical breakdown. We start from one cluster, go to 2, 3, 4, 5, but it's always you're getting these subdividing of the clusters. Or perhaps looking a little bit more prettily at it, what we can see is that the clusters are breaking down as we move sort of from these more complex descriptions, we can see that we get this tree-like structure with sort of some sort of non-tree-like behavior at the very end once we start running out of data. So we get this hierarchical description of data, which again agrees nicely with a lot of these types of theories. And the hope is that this can generate some notions of how we can generate long time scale structure in data. And how we've been thinking about this more recently has been kind of with this kind of philosophical picture in mind of if we can think about an abstracted behavioral landscape, if you will, where you have a little thing which is moving from place to place, and you have this is your behavior, and then each of these basins might be a different stereotype behavior, and then we're hopping between these different basins by having some deterministic component and a stochastic component that's based on that. And so this is very much inspired by the picture that was derived from by Greg Stevens and Bill and Will Rue in their previous work thinking about C. elegans behavior. But the thing is, to get non-Markovian dynamics, we can't just have a picture that looks like this, because unless we have something really interesting happening with how the temperature is changing as a function of time, we could only generate Markovian dynamics with this kind of hopping between basins and basins. So what has to happen is we have to have this overall basin shift with time, moving around and changing the relative interactions between these sets of things, between these individual stereotype behaviors and how they move between them. And that can be generated by changes in internal state or changes in sensory state. And this notion of how that basin is warping and how that dynamical picture is changing is really what we mean by internal states. It's how are the relationships between the behaviors changing from what we would naively expect from marginalizing across all time. And so in sort of a first pass from this, as I sort of noted, we can try and write down simple models, and it's really hard to generate dynamics like this. So what Atai, who's a former postdoc in the group, and Catherine have been doing is they actually we said, okay, we give up. Let's just try and completely over-parameterize the problem, and then we will try and make it simple later. And what we do by that is, what I mean by that is we just essentially train a recurrent neural network and treat that as a dynamical system model for the data. So over here might be sort of a one-hot encoded measure of the behavior. So this would be one, if it was just behavior 1, this would be 1, 0, 0, 0. If this is behavior 2, it would be 0, 1, 0, 0, 0, and so on. We train a fancy network to then predict the next state. And then we have probabilities of what the next state is. And then we use those probabilities to select the next state, which then gets fed back in. And then we can have this dynamical model, which has a huge number of parameters, but maybe we can still learn something. In a similar manner to, like, for example, the Ising model work that's been done, popularized by Bill and others here, thinking about how you can fit a whole huge number of JIJs, but really by understanding the structure of those JIJs and really sort of delving into them, maybe by taking a look at sort of the structure of the underlying properties of this sort of crazy dynamical system we've built, maybe we can learn something. And I'm not necessarily going to claim I'm going to show you we learned something today, but we can thus at least think a little bit about some, hopefully be inspiring some new ideas that people can think about for this. So long story short, we can reproduce the types of structures we see in the data. It's not a perfect quantitative agreement. So here the solid lines of the data and the dashed lines are what we get on average for the RNN. And so we can indeed get these similar types of long time scale dynamics. Moreover, we can also reproduce a hierarchical breakdown more or less of the space. It's not quite the same. Again, it's not perfect, but we can generate a lot of the similar types of summary statistics that we were hoping for. And given that, we can then play a bunch of games. Like we can actually isolate where the fixed points of this network are. So where are the points where if we kind of eliminate the noise by saying, okay, let's run the system where instead of I'm going to pass around something that I've sampled, I'm just going to pass back around the probabilities of which state I'm going to go to next. We can then see where it settles down in because the system winds up being dissipative. And we get a bunch of fixed points, which are and these are fixed points which are in this complicated sort of set of internal states within the recurrent neural network. And if we do that, we basically can see each of these, if we start from what each of these original behavioral states, there are these numbered points in here, and then we let it settle down, we can find where those fixed points are. And if we do a simple multi-dimensional scaling, embedding, or pick your favorite dimensionality reduction approach, these are what sort of the structure of those fixed points look like. And note how this method didn't know anything about the space other than the fact that it was, these were sort of what was generating overall sets of 1s and 2s and 7s and 117 values that are sort of the state numbers. And if we do that, it rediscovers the exact same sort of structure that we see. So locomotion, locomotion, posterior, posterior, slow, slow, idle, idle. So this network is kind of recapitulated the space. Also, this was kind of nice because it sort of showed us that maybe this space is actually giving us a representation which is dynamically interesting. And then we can starting from each of these fixed points, then we can start playing this game that we knew by thinking about Langevin dynamics, where we have this deterministic part where we take these probabilities and feed them back around near the fixed points. But now we can feed them back around with a little bit of noise. And we can vary that amount of noise and then see kind of what happens with the network. And turns out we actually can see that average over all time, this network actually looks surprisingly well like a set of basin hoppings all at a constant temperature by measuring the escape time it takes to actually get out of the system, get out of each particular well and move to a different behavior. And so this thus means that average across all times, we have this constant temperature, but what must be happening is these various basin heights must be changing. So that's something that we've been thinking a lot about, and how sort of this context might be changing as a function of time. Again, this is sort of half baked at the moment, but I just wanted to show you guys who are thinking about kind of where this is moving forward on this. And again, please interrupt me, ask questions. It's a little disconcerting to have a wall full of videoless individuals not responding. So please let me know if there's anything that I can clarify or anything you want me to add on to.
So Gordon, let me try something to respond to your invitation. I guess the question is whether are the states an emergent property of the network, or are they? You haven't hand coded the attractors, right?
No, we've only hand coded the basically the input and output. So we basically said there's 1 to 117 there, and there's 1 to 117 there, and the order is arbitrary. And the network has how many degrees of freedom? The network has, so measured in terms of parameters, I think. No, in terms of dynamical variables, I guess. I'm a little. Maybe that's right. Sorry. In terms of dynamical variables, this one, the way we wrote it here, would have 256. Okay. So I guess the question is, so in some way, in order to get the right, there's this, in the old discussions about 1 over F noise, right? There's the contrast between something that's truly an emergent property of some many-body dynamical system, and alternatively, there are things like what happens for conductance noise in semiconductors, where you have basically individual carriers get trapped, and there's a distribution of trap depths. But your situation is interesting because it sounds like, well, the way you get this very wide range of timescales is that you have states, and the time to escape from the states is broadly distributed. But of course, the states themselves are emergent properties of dynamics, and therefore so are the trap depths. But so that's one confusion about how do I, is this a collective effect or not? And then the other point is it really true that if I look at the data, the distribution, because the long timescales don't come from the escape from a single state, right? They come from escape from some neighborhood of states. Yeah, that's exactly right. So like I think that second point, I think sort of whether it's well, I'll try and do the first one. So asking about whether it's a collective sort of emergent property or not, I don't necessarily know if that analogy works super well here, particularly because it's, we're looking, the dynamics that there's two sort of full dynamics. There's the dynamics of like hopping from state to state to state to state. And then there's the dynamics of what's the underlying kind of tilt of the overall kind of washboard, if you will. And I think, and that gets to your second question, which I think, and that exactly is what most of these models are starting to suggest, is that in fact you actually are, I mean, and this is exactly what our analyses and other things showed previously, is that you have to have a hierarchical arrangement of the washboards. And sort of how we've written down a more simplified model that's not quite ready for me to talk about in public yet, because I'm still double checking some things, is that we essentially write we write these internal states as kind of a series of sort of as a matrix decomposition, where you have your mean transition matrix, and then you kind of have these block diagonal kind of hierarchical structure transition matrix, which where basically essentially your transition or your sort of your trap depths are covariating together. And they're covariating together all of these different scales where you have stronger covariance between like moving, not moving, and then sort of within sort of regions between moving and locomoting, you have different sorts of things. And so that's kind of how we're trying to build this up. Again, I'm showing this, this is one of those things of risking showing something which I don't think is quite done yet, but I just sort of wanted to show something which is a little bit new.
Thanks. No. This was a big help. You should go on. Unless other people
Unless there's other questions people have.
Gordon, I had a quick question. This is Chris Lynn, by the way. So just to go back a little bit, you when you were talking about the information bottleneck with the different sort of behavioral states, you mentioned that there is this hierarchy emerged as you vary sort of the accuracy of the clustering, or the fine grain.
Well, either the time or the descriptive.
So my question is, so did you build in the hierarchy into the way you were finding the clusters, or did it just emerge that there was a hierarchy?
No, so we didn't build it. So this is not a hierarchy clustering. Now the information bottleneck sometimes, and sort of the work from may he rest in peace, Naftali Tishby and others, looking at basically the what the structure of information bottleneck is learning as you change representations. You do tend to get this hierarchical structure that emerges, but that's a result of the particulars of the cost function, not because you kind of put it in by hand. The other thing which is not in by hand is the fact that these clusters are contiguous, right? The fact that they're contiguous in the space, like this algorithm knows nothing about that. It's just like I could reorder my list of things in any order and that would still work and it would still return these things. So you could argue a little bit about whether a hierarchical structure was guaranteed to come up at all, but the fact that it's contiguous in the space is the bit that sort of says, oh, that actually means something.
Yeah, that's interesting. And I might have missed this, but what are the two dimensions in this space that you keep showing? I might have missed that.
Oh, in sort of like in these behavioral maps?
Yeah.
They're arbitrary. So it's a nonlinear embedding. So it's basically everything is related to its relative position to everybody else. So the whole thing could be completely rotated or translated in any way, and it would mean exactly the same thing.
And
unfortunately, like I can't have like axis 1, axis 2, which is why I violated one of my prime rules that I always told to my students, and there's no axis labels on this.
Does sort of the contiguity of the clusters change if you look at different dimensions? I mean, when you say contiguous, do you just mean visually it looks contiguous when you project it onto these two dimensions?
The regions are contiguous. Basically there is they are, I guess, not simply connected, but they're all connected to each other, right? So
cool.
I'm sorry, was there a? I know that's right. And if you go up to 3D, basically, you can show that if you get the 3-dimensional embedding is basically the same thing for two-dimensional embedding, just sort of wrapped a little bit. So
Cool.
because of a bunch of reasons, we find that particularly for flies, that 2 dimensions really is sort of captures all the structure here. Again, we could have done a clustering without the spatial structure, but I think the spatial where we just actually cluster high dimensional data, which in some ways all you care to do is cluster, you're better off always just doing that in the high dimensional space. However, this allows us some more interpretable frameworks to build because we can start to see like how these various things are related to each other more nicely. Now that's kind of the logic and always the trade-off one makes when one's trying to build these types of representations. That having this, building it off of the spatial structure gave us a bunch of advantages which overcome some, perhaps some of the other problems that could have been there.
Cool, thanks.
Other questions? All right, so what I want to do is sort of thinking about this context dependency. I want to go back to thinking about the descending neurons again. As I promised they would show up again. And so what you can see is one of the things that we found here is this notion of context dependency in behavior. So what do I exactly mean by that? So here is this one. This is a really nice clean line. You can just sort of see this pair of neurons, one on each side of the fly, and you're going to excite the fly as it's behaving around, moving through. And what you find is you actually get 2 statistically significant regions that emerge, not just one like that I showed you before, where you see the head grooming. You get this big region here, you get this little region over here, right? So what we can do is we can say, all right, given that the fly, when we turn on the red light, starts doing this behavior, which is sort of like little leg twitch things, what was it doing before? So if you ask that, what you see, this is this probability distribution, what it was doing in the 2 seconds before that we turned the light on. And we get the sort of thing which is spread out throughout most of the space. Alternatively, we could measure the density before transitioning to this tiny little region here, which, as it turns out, is sort of this wing flicking behavior. And if you do that, you get a much more isolated region here where you get basically wing grooming type behaviors and every once in a while sort of a resting behavior that shows up right before. And so, and you can measure the mutual information of given where I was before, which of these 2 states I'm going to go as. Obviously, I can get up to 1 bit, and we measure 0.5 plus or minus 1 bit. So you actually do have information about which behavior you're going to go to next. So this is this notion of context dependency. And if we measure that, that actually shows up multiple times. So in about 85% of the lines, so actually most of the lines showed that we would have multiple excited regions. That's part of why I showed you that, the one previously, just because it was so clean. Most of these regions all have kind of, if you're below this red line, that means you have more mutual information than the control flies did. And if you're above this line, that means you're above 0. So you can see the vast majority of things are kind of within this wedge of the space. So we see this context dependency, which is showing up over and over. And so what might this have to do with long time scale transitions? And so what this is related to this idea that, as a matter of fact, if you have a hierarchical structure, performing this, you might want to encode your behavior in a way such that if I'm in the middle of one task, if I'm in the middle of building versus courting, I might want to kind of rewire my neural circuitry, or in something which was shown beautifully by Gwyneth Card's group, if I'm walking instead of flying, I'm just going to completely revamp how I'm going to code stuff down this information limited channel. And I won't go into details. And then what we eventually sort of wound up showing, and this is some work by a bunch, actually with a bunch of students that started as a project from the Boulder Summer School a couple of years ago, really showing that if you take a really simplified model of behavioral commands with information bottleneck, you can wind up showing that this type of context-dependent structure winds up leading precisely to these types of predictions where the same neuron might be encoding different things if you're trying to optimize your ability to command as many behaviors as possible through an information limited channel. So I encourage you guys to take a look at this. And this was sort of nice because it also wound up agreeing with some of the neuroanatomy results from Gwyneth Carr's group as well that were published a little bit. And the hope is that we can try and find interactions between this sort of long time zone structure and the neural dynamics. I think that really is going to be the key for understanding kind of relating behavior and the neural structure underlying its generation, is basically because of the fact that the behavior really is such an important thing that the brain is doing. That context and that structure of the behavior is going to be important. And if you're just kind of averaging uniquely across all time, rather than trying to look at the time-to-time fluctuations and variance and dynamics that you're seeing in the actual state of the animal, you're going to wind up missing stuff that you're just marginalizing over everything. So this is kind of a break, but if there are any questions, feel free to go ahead. If not, I'll fall forward. But so the next little bit of the talk is thinking about some more recent work that we've done, thinking about really trying to focus on variability, not just across time, but also across species. And as it turns out, those two things wind up being surprisingly interlocked with each other. And that's sort of as a result of this recent paper. And thinking about evolution of behavior, we run into a lot of these questions of variability that we saw previously, and that people have studied a lot about how genes evolve by measuring sequences. We have this nice way of doing things for that, and measuring sort of change, and which then lead to changes in neurons and then development. And of course, these things are all linked to each other, and all of this stuff kind of is linked together to drive changes in behavior. So as a result, trying to link these things to these things, so trying to link the genes that are evolving that result in evolving behaviors has often been complicated because there's kind of this sort of opaque wall which often sits between the two. And there's two kind of ways that people might have thought about this. One is you have these candidate genes. Genes that people know that if you knock this thing out, you cause big changes in behavior. One example, this is the fruitless gene in fruit flies, but however, often in cases where often we take these nice candidate genes, often it doesn't wind up resulting in that much insight. So for example, well, it can result in insight, but not necessarily the insight we hoped for, I should say. This is some really heroic experiments that Jessica Candy performed, where you basically take fruitless and double sex, which are these really long, pain to deal with genes, and try and actually move the copies of that same gene from melanogaster into other species. And it turns out these genes are heavily incorporated with courtship behavior and other things. It turns out if you move the behavior, you essentially don't, you can move this whole gene from melanogaster into another species, and it doesn't change the courtship song of the animal at all. So it turns out in this case, and in many other cases, this candidate gene approach, where we just sort of think really hard and make a really good guess, doesn't tend to result in the thing we hope for. Similarly, what we could do, and this is again another, so you'll notice my many featuring of Jessica Candy's work here, because it's always this really careful, crazy experiments that she's done. And then you could then look at, okay, let's look at wing rowing, which is this very descriptive sort of behavior where one species, Santomea, does these wing rows out like this, as you can see here, and this other species, Yakuba, doesn't do it at all. And they're both very closely related, about 400 kiloyears apart. And as it turns out, you can try and do QTL analysis, where you're trying to isolate genetic markers that would relate to this change in behavior, and you only get these sort of really big, wide regions of the genome. And it's harder to, and it gets really hard to really sort of narrow it down. You can build these targeted integration lines, and it turns out not being super helpful. And so, or at least generating or being able to narrow it down more. So where we came into this and where we were thinking about this was that maybe the problem is that we're looking at focal behaviors. We're looking at these individual behaviors that the animals are doing that are that look like they're evolving, but really what might be evolving is there's some upstream regulator. And we have some notion this might be the case from from David Stern and other people's work looking at cis-regulatory elements in morphological evolution. So the idea that there might be some upstream regulator that's regulating a bunch of behaviors at once, and this could result in changes in kind of neural connections and other things, might be something we could look at. And this is the type of approach, type of thing that our type of approach might work well for. So we took data from these 6 different strains of flies. Again, this is Jessica's experimental work. And it's wound up being about 600 flies from 6 species and then 19 strains, because we took multiple strains from each species just to make sure that we weren't fooling ourselves by picking just one strain. And then Damian and Catalina, then they played our usual game that you've seen. They build a behavioral map, and then they really tried to understand if we can understand the variance between these species in a rigorous way, trying to understand how these different types of behaviors are co-evolving together. So we can make this map. It has the usual bits and pieces of idle, slow, locomotion, abdomen, posterior, anterior. And it has the nice transition structure that looks similar to the types of maps that I showed you before. And in fact, we also get the same type of information bottleneck results where the space breaks down hierarchically again if we play that same sort of game. So we have the map, but this is of course all 6 species averaged together. And so what we want to do then is we want to break them down into 6 species. So here are the 6 species we had: Mauritiana, Melanogaster, Santomea, Seychellia, Simulans, and Yacuba. And you can see just quickly looking at the mean behavior that there's pretty wide differences between the species. But the question then becomes, is their behavior alone a trait that we could try and kind of map and make some sense out of? And so one way to try and convince ourselves of this is just trying to do a classification problem. We can take the maps from each of these roughly 600 individuals and just compute distances between them by computing, say, a Jensen-Shannon divergence, and then putting them onto using t-distributed stochastic neighbor embedding, or UMAP. This particular case is t-SNE, but you could use other things, and just putting them on a 2D map. And what you can see is it's not random. It's not perfect, and not everything is perfectly separated from one another, but each of these different species are more or less separated often, and each of the symbols are different strains. But you're also not seeing separations from the different strains of the same species. This is just a visualization, and it's also only a very low dimensional visualization. Let's actually take the full data and ask our question of what's the, how well can we do a regression to figure out which one is which? So we just did a very simple logistic regression between the 6 different things, between the 6 different species. Note how this isn't like some fancy neural network. This is just simple multinomial logistic regression, and we're getting about 90% on average accuracy. And this is test set accuracy between them. We get about 100% training set accuracy. So basically, what you can find is that we can generate to about 80% accuracy which species is which, just based on the behavior it's performing. And we have a bunch of other reasons to believe it's not just because we had some experimental changes or other things like that. So based on that, this is really the work of Damian Hernandez and Catalina Rivera, who really sort of spearheaded the analysis side of this work, is they fit a generalized linear mixed model onto this tree. So basically what that means is you fit a mean, so you fit an ancestor behavior, and then you fit 2 covariance matrices. So one covariance matrix is how are behaviors co-evolving together as you move along this phylogeny, or doing a random walk as you move through. And then the second question you ask is, if I sample at a random point along the tree, what's the probability I'm going to get a covariance between those 2 behaviors? That's our individual covariance. So that's basically so what this is nice about this is we've separated out these 2 types of variance in our data. One type of data is this data of how behaviors are co-varying together, and the other type of variance is how, if I was to pick two flies at random from the same species, how would I expect their behaviors to differ, and which behaviors would differ together? So what we could do, we fit that map, so we wind up getting a plot like this. We get a bunch of ancestral states in the middle, which is sort of fun, but these aren't super testable hypotheses because there's not a fossil record for behavior, unfortunately. So, but the real focus here are going to be on these two covariance matrices. So the first one is individual covariance. So for the individual covariance, what we see if we plot that covariance matrix like this is we get it's pretty modular. In fact, if we were to cluster it by sort of this 4-cluster solution, we see something that looks sort of like the bottleneck clusters we got before, which is convenient because it turns out that we actually compute the bottleneck clusters. Here, we get almost identical structures, not quite the same, but it winds up being really, really close. So these two things, which are generated completely different ways. One is looking at behavioral transitions, and one is looking at this fit along the phylogeny, generate almost identical results. And our interpretation of that is that there's sort of an ergodicity in a lot of these behavioral measurements. And what I mean by that is averages across individuals is similar to averaging across that same individual over time. So basically, you measure a bunch of these individuals, and you're going to get, for some relatively short amount of time, and you're going to see biases which are related to these long-term individual states or within regions. And if you would get similar sorts of structure measured one animal and measured over time. And the consequence of this, particularly when thinking about the focal trait idea that I talked about before, where we say, let's look at one behavior that is clearly different between these two species, and let's try and correlate genes with them, is that means that measuring the relative frequency of that trait is then going to be highly affected by these internal states. So you need ways of marginalizing across it. But one of the things that, so we did this. So one way of trying to do this in a simple way is you basically say, you know what, like, let's take these bottleneck regions over here, maybe add a couple more to say to a 6-region state, and say, you know what, I don't really believe that these species are going to have different amounts of time they're going to spend grooming their head. Right? They're all kind of grooming their head the same way. And really what I want to care about is, are there different ways they're grooming their head, or the different types of things? So you just normalize across sort of these regions. So you equate to make sure that all of those regions stay the same. And then you basically ask to see, have my results changed? So one of the ways you can ask is you can measure your distribution of any of these given behaviors or before normalizing for 2 species. So this is for Drosophila santamea and Drosophila yacuba. And before normalization for, say, these 3 red points up here, what you can do is you can measure the Mahalanobis distance, so the difference in means divided by the sum of the standard deviations and quadrature. And you get something which is very small, but after you normalize, you can actually see that you get a variance between the 2 species in terms of their relative frequency of this trait. And you can also get the reverse, like in these blue points, or you can get it staying roughly the same, like in the black points over here. So this notion is basically that if you are going to do the focal trait thing, you need to actually measure multiple behaviors in order then to make sure to normalize across all of these, or at least to marginalize across all of these hidden states that you might not have seen. But what we got really interested in was then looking at the phylogenetic covariance matrix, which is basically saying, how are these behaviors covariating together? And if you do that, what you basically see is now you get something entirely different, where now you get these, you still get something which looks modular, but now these modules look like sort of disconnected regions of this behavioral map. And what we then propose is then what we can do is then look at the eigen, because it's a covariance matrix, we can effectively just do PCA by looking at the eigenvectors and values of it. And so we get a set of, say, our leading eigenvectors in the data, and then we can do projections on them. And this gives us predictions of kind of multi-behavior traits that we think might be evolving between these different species. So a couple of important caveats, I think, is that, first of all, this is only on 6 species. And so it's possible that some of this modular structure that we're seeing, particularly in this matrix, might be the result that we're undersampled along the phylogeny. So that's something that I think is an important thing, and something that we're kind of building out for in sort of a couple of different experiments we're thinking about in the future. So are there any questions sort of on that section? I only have one more sort of speculative bit left. after this. So we'll there. And so I just want to end kind of wrapping back up to that sort of silly picture of the prairie dog conducting I had at the very first slide, thinking about themes and variations. And really like this notion of trying to, even if just metaphorically, thinking about behavioral, our structure of behavior as sort of this sort of dynamical flowing sequence of states that are governed by how internal states are modulating the landscape beneath it and the amount of noise which are fluctuating on top of it. And almost trying to think about what might the right metaphors or what might the right structures be as we try and build things out. And the thing that comes up most often, if you look at the literature and behavior, is notions of language. We can see this over and over. Just to pick a few particular examples, you can see that the words syntax, grammars, all of these things come up often when people are talking about trying to model sequences of behaviors. Now, granted, almost all of these things are talking about behaviors on relatively short timescales. Again, on this timescales of order of second or a couple of seconds. And how do we then, we could try and ask ourselves whether sort of this notion of language is sort of a right thing. And moving more to the philosophy of the thing, the worry I sort of have when thinking about language as the right metaphor for this type of thing is that it's almost too rigid and short time scale. When people think about syntax in particular, maybe if you can find some magic way of writing a mathematical framework to understand how you go from sentence to paragraph to chapter to book, which there is some degree of this, but it's most of the effort on understanding these things are on relatively short timescales, even with the types of transformers and recurrent neural networks that are used to generate bits of text. Maybe poetry has the same sort of structure, but there's not kind of a mathematical framework that underlies it. So where my brain kind of goes, and the type of theory, right? Goes to this question of what type of theory do you want to have when you're thinking about a system? The type of theories that I think about, and maybe this is my own bias as a incredibly mediocre jazz pianist, is thinking about kind of music theory and the structure behind music. So as you look between these different types of, there's different, for example, changes in key signature, which are all related to each other in very careful ways through diatonic scales and notions of nearby chords from this. But you can also build structures like over a symphony where you have overall moving from exposition and development to recapitulation, and people have developed mathematical frameworks to build on top of all of this. Just skip this. But the idea then can become like, and even this sort of notion, so just stealing from this sort of cool, this site that I think made a bunch of interesting visualizations of Einstein on the Beach, the 5-hour-long Philip Glass, Robert Wilson opera, how you can basically build these type of long timescale structures out of shorter patterns that go and build motifs. In the same way, you can build 15 hours of music by listening to Wagner's The Ring Cycle, where you can see leitmotifs that appear and disappear and move in and out. And there's a sort of living and breathing air that goes as a result of these overarching structures, and people develop mathematical models. And these are sorts of things. So something we've been starting to think about this connection a little bit more literally, but more figuratively, we've also been sort of thinking about how to build these type of long time scale or almost tonal structures into building multi sort of time scale representations. And this is some of the work that Kanish Jain's done that I don't have a huge amount of time to talk about, but I think that could be this is sort of how we're starting to think about these types of problems. And with that, I just want to thank all of the folks that actually do the work with me, great group members, and tremendous collaborators I've had the opportunity to work with, and people that give us money. And I will also shamelessly plug that we're currently looking for various folks to go work on these sorts of things. So if you're interested, just let me know. Thanks.
Thanks, Gordon. That was great. We already have one question in the chat, and so let's work on that one while other people collect their thoughts. So the question is, what share of the differences in behavior that you observe between these species can be simply and simply is in quotes, attributed to the differences in body size and morphologies.
Right. So the reason why flies are kind of nice here is that their body size and morphology are actually pretty consistent, and we can use methods to kind of coarse grain over them. In fact, some of the fancier stuff. So I can confidently say here that the body size wasn't the effect, because we can see the same animals performing similar behaviors and they map to the same part of the space. And that wouldn't be the case if we were really worried about the fact that it was there was a morphological component to it. When we sort of are doing a little bit further afield than some of these flies look, because all of these flies here in the Drosophila melanogaster species subgroups, they all kind of look similar. They're not identical, and you can tell them apart if you know what you're doing. But but as we go a little bit further afield, we've had to do a few more fancy tricks to come up with, basically to describe their posture in a lower dimensional space where everything is green. So I can say for these data, the morphology has very little effect, but for other things, we worry about it more.
Good. Are there other questions? Maybe the people who are most likely to ask questions have been following what you've been doing. So, so for our third talk of the day, as promised, we were going to go all the way from behavior on the scale of large numbers of organisms, individual organisms, to thinking about its neural basis. And the example that Mark's going to focus on is one in which I think it's fair to say that over decades, the quantitative analysis of behavior really guided the search for neural mechanism in a very explicit way. And so I think it's a great example on which to end the day. Another thing worth pointing out is that we have finally succeeded in having this be jointly not only an activity of the ITS and the Center for the Physics of Biological Function, but it is also jointly the weekly seminar of CUNY's graduate program in neuroscience. So it's great to welcome that set of colleagues. We all finally converted our good intentions into actual coordination. It's always hard. Mark Goldman from the University of California, Davis, is a theorist who's worked on a wide variety of problems in neuroscience, from the very molecular to the very systems-oriented. He's going to be telling you about a great chapter in his own work that's occupied it for quite some time. Rather than reaching back and telling you about things that he did before, I'll note once again this sort of coincidence that today all three of our speakers have been very involved with education. Mark obviously has done his usual job at UC Davis, where he's also played a big role in building a more quantitative biology program. Work that's recognized by his being a Howard Hughes Medical Institute professor. And he's also served as co-director of the summer course at Woods Hole, through which many students in our field have passed. So it's a pleasure to have him here. And Mark, please.
All right. Well, thank you, and thank you for inviting me to this really fun event. So I'd like to sort of switch gears, as Bill said, to talking a little bit more about the neural circuit mechanism level of behaviors. And just to start, here is sort of my cartoon of what is the, what do we all think of as two key ingredients of behavior? Of course, stimuli that trigger a behavior and the responses. But what I want to focus on here, which still grossly, obviously, downplays all of the intricacies of behavior, is that between stimuli that trigger behavior and the responses thereafter is the accumulation and storage of information within the brain. And in particular, this issue of accumulating and storing information is thought to underlie much classic work in experimental neuroscience, for example, on decision-making, where the basic theory of decision-making is that animals accumulate information over time, potentially short time scales or very, very long time scales before making a decision. as well as things like navigation, where we know that animals can accumulate velocity signals to maintain a an internal state representing their position in the world. And I'll talk about some other tasks where this accumulation and storage occurs within motor systems. But before doing that, I wanted to set up the big picture and sort of why this has been a problem I've worked on and many others have worked on for many years as theorists. And the problem, I think, is most simply stated in this cartoon, that in many memory and decision-making circuits of the brain, we see the accumulation and or maintenance of signals for order 1 to 10 seconds. So here it is just a cartoon of a stimulus being presented over time, and the neuronal activity just representing the overall activity of the network is accumulating the stimulus, and then at the offset of the stimulus, storing it in working memory. And I'd like to note that, intentionally I've represented this accumulation as an analog process, because I think the real challenge is not how something is maintained as a 0-1 state. That's very easy, but how analog memories are maintained and how we can maintain what at least appears to be graded signals with an analog value, or approximately so. So the big puzzle is that most neurons, when one looks at the componentry of them, that the dominant timescales of most components at the neuronal level or at the synaptic level sit on the tens to hundreds of milliseconds timescale. And that's an order of magnitude shorter than what we think of as than what we observe in the timescales of neural activity. And that leads to the question, what bridges this gap between the intrinsic timescales we see on neurons and synapses and the timescales we see on neurons? So I'm going to start this out, I think, as Bill was alluding to, with a particular model system that I've worked on for many years. It actually, the credit really goes to, people like Bob Baker and David Tank, who really sort of pushed this system, and particularly David, as a model for short-term memory. And it is the eye movement system of any vertebrate animal, but I'll show you some older work from David's lab in terms of the data I'm showing you from the goldfish. And this is the basic behavior I'll be describing. So you'll see a zoom in on the eye. And then I'll explain where the memory system sits in this behavior. Okay, so here's the basic behavior, a very simple one. So you just see that animal is holding its eyes fixed and then makes rapid changes in where its gaze is directed. Those are called saccades, those rapid changes. And what I'd like to point out is, though here you're looking at a movie in the light, all of the data I will show you is in the dark. So I'm not going to be showing you the effect of a visual stimulus, but rather internal dynamics in the brain that enables the animal to hold its eyes at various positions. So here's just a cartoon, some data and some cartoon of what you just saw in the movie, just showing eye position over time. And again, for the fish, they tend to hold their eyes in one location, then make saccades, change to a new location, and another saccade, and another fixation. What commands those changes in position are a set of neurons that we describe as having a brief burst of neural activity. And there's two populations, one which you can think of as excitatory and directing the eyes one way, one you can think of as inhibitory and driving the eyes the other way. And if we look at that, these neurons are only active when the eyes are moving. And in fact, their activity is very tight. If you plot it together with eye velocity, it's very tightly related to eye velocity. So we say that these are eye velocity encoding neurons. It is it's been known forever that muscles are spring-like and can be described by Hooke's law to a first approximation plus some viscous damping. And of course, to control a spring's position, you need a position signal, something that is the force proportional to the positional displacement. And it was therefore known that these eye velocity signals would not suffice for controlling eye position. They would lead to a rapid movement, and then the spring-like musculature of the eye would return the eyes back to a neutral position. So that immediately led to the idea, and David Robinson was the first one to really sort of flesh this out in terms of laying out models, that there must be a neural integration, something which converts these velocity signals to the ultimate eye position controlling signals that keep the tension on the eye muscles. And those neurons were identified. And here I'm showing a recording from David's lab and in the goldfish. And what you see in these neurons is a brief excitatory input increases the firing rate of these neurons. But then after the offset of that, there is persistent neural activity in the absence of the driving command. And again, the lights were off. So this is all internal. There's no external feedback through the world. And this persistent activity you see increases as the eyes increase their position and respond to and keep on responding to individual excitatory or inhibitory pulses, such that they're storing the running total of input commands. That is, they're performing an integral, hence they're called the oculomotor neural integrator neurons. This is actually showing an eye trace, but for the rest of this section of the talk, I will just be showing you what are called tuning curves. So each dot on this curve is showing the maintained firing rate, that is the number of spikes per second produced by a neuron when the eyes were at a particular position. And this is typical of the neurons in this system. They have a threshold as the eyes are moving from left to right. They have a threshold at which they begin firing, and then they have a quite linear relationship between firing rate and eye position. Of course, here I'm just showing you one neuron. And if we were to look across the whole circuit, there are many neurons. There's actually in the fish only order 100 neurons, so it's a relatively small system. And the different neurons, as shown here for neurons recorded on the right side of the brain, they have different thresholds at which they begin firing and different slopes. And these are just fits to neurons, but I think are a good descriptor. Now, the truth of the matter is this is actually a compilation of right side and left side neurons, because statistically, the neurons on the left side are mirror images of the right side neurons. So the data were just combined, but this is the basic pattern you should think of. Right side neurons increase their activity when the eyes move from left to right. Left side neurons decrease their activity when the eyes move from left to right. And the nice thing about this circuit, and we're still, Sebastian Sung at Princeton, has actually just completed the connectome of one side of the integrator and is working on the full connectome. But basically, we think the gross architecture is made up of 4 populations of neurons divided by the midline of the brain. And those populations are divided into excitatory neurons and inhibitory neurons. And there appears to be a very simple pattern of connectivity, that the excitatory neurons project to the same side of the midline, and the inhibitory neurons project to the opposite side of the midline. What this means more functionally is that there are recurrent excitatory feedback loops within each side of the brain. And then potentially there are recurrent disinhibitory feedback loops between the two sides of the brain. That is to say, if you wanted to go from a neuron on one side of the brain back to another neuron on the other side of the brain, you'd either go through recurrent excitation, or you'd go through a double negative form of feedback where the activity goes to one side of the brain through one inhibitory connection and comes back with inhibition to the other. So I'm not going to get into some of the details, but you can find some of the details in papers from David's lab and my own lab. But what I do want to focus on here is just the basic form and the basic mechanism here, because the form of these feedback loops describe the traditional mechanism for how persistent activity, memory storing activity, is maintained across many brain circuits and across many tasks. And that's shown here. So basically, simple story, a brief transient command input comes into a neuron or a neuronal population, a typical isolated neuron. If not connected to anything, its firing rate would come up and come down on some neuronal time scale. But since that neuron is connected to others, either through recurrent excitation or through recurrent disinhibition, the neuron can maintain its firing rate. So if these are neuronal populations, when certain neurons increase their firing rates, they can increase the firing rates of their neighbors, or they can decrease the firing rates of the neurons in the opposing population that were inhibiting them, thus removing inhibition from themselves. When this is tuned correctly, and that's something I'll get into, that can lead to persistent activity that is maintained over long time scales, much longer than the intrinsic time scales of the neurons. All right, So I just also want to note that this is actually a figure here, which actually is related to a more cognitive memory tasks. The experiments from Rodolfo Romo, and actually this paper was a beautiful science paper from Carlos Brody, your colleague at Princeton, and Christian Mawkins, who was his postdoc at the time. All right. So again, key features here are recurrent excitation and recurrent disinhibition. And together, these are both positive feedback mechanisms by which activity is maintained. But we wanted to push this and say, okay, so these are grossly speaking positive feedback mechanisms, but how can we think about the system and collectively how the interactions drive the system? And can we also come up with more geometrical pictures, like energy surfaces or attractor landscapes? So that's what I want to show next. So here is just a simple cartoon of activity of two neurons on the same side of the integrator. So again, each neuron on the same, would be on the right side. As the eye position increases, these neurons increase linearly in their firing rates with eye position. So on the right, what I'm showing is plotting those two neurons against each other. And here we just see that this is the pattern of activity among those two neurons. And you can think of the saccade as taking you from one pattern of activity to another pattern of activity, and the eye position is encoded along this pattern of activity, which in general is the full n-dimensional one, and I'm just showing in 2 dimensions. And that led Sebastian Sung a number of years ago to describe this system geometrically as a line attractor. So the idea here is that this set of patterns of activity here, that is representing an eye position coding axis. And when activity departs from that coding axis, the dynamics of the network form a line attractor. That is, activity is returned to this line, but any of the analog range of firing rates along this line can be maintained. So this is a line of fixed points, or a line attractor. And I just wanted to make that concrete with this fun movie that comes from Sebastian and Dan Lee and David, just actually showing you the dynamics of this line attractor. Marvin the Martian is actually, as Marvin's eyes move, those are the eye movements of the goldfish, and you'll hear the crackling of the action potentials of a neuronal recording as you watch the state of the system move back and forth. And again, you can see right there where the activity left the line, but then was attracted back to it. You also get a sense for the noise in the system, something I won't talk about today, but something we've also worked on. Okay, and you get the idea. So to explain this line attractor, the sort of simplest models, and these were the first models that Sebastian made prior to my time actually working with him as a postdoc, were just saying, let's look at a linear network. So the neurons we saw have very linear relations to eye position. If we ignore the thresholds, which may or may not be a good idea, we can just come up with a simple linear network. And that would be to say that, the changes in firing rate have an intrinsic decay on a timescale tau neuron, plus neurons are driven by the activity of other neurons through a linear weight matrix, just a linear interactions defined by a weight matrix Wij, which is the strength of the connection from neuron J onto neuron I. And then, of course, there's external inputs like the saccadic inputs to be integrated. So, standard calculation from linear algebra, the eigenvectors of this set of equations represent independent patterns of neural activity. And those patterns of neural activity will have their dynamics determined by the associated eigenvalues. And in particular, for any eigenmode, the time constant associated with that mode is the time constant of the neurons themselves divided by the, -1 plus the eigenvalue of the weight matrix. So here, I've sort of kept separate the -1 corresponding to the decay term plus the Wij corresponding to the recurrent connectivity, because that gives you just a nice sort of concept that the idea for getting a long time scale in the system is to have a leading eigenvector of the weight matrix with an eigenvalue of 1, where 1 defines, if you wish, the scale of decay in the system. So if the feedback can offset the decay, then one can get an infinite timescale mode or a line attractor mode. And that, in the pattern of neural activity corresponding to that would describe the patterns that you saw in the experimental recordings here, or in the theoretical picture of the line attractor. And then these attracting directions, these would correspond to to eigenvectors whose real parts have eigenvalues less than 1, so they correspond to decaying modes. All right. Okay. So we wanted to take this system a little bit more seriously and go to a nonlinear network, which described it. So we made a population model of, a network model where we made 4 populations, each of which had 25 neurons, representing the order 100 neurons in this system. And again, it should look very similar to the linear network model, but basically we said that the firing rate changes. There was a decay in the absence of any external input due to intrinsic leak components. We actually fit a biophysical model to capture this function f. I should say we actually built a spiking model with the full glory of ion channels and things, but the essence of it really can be described by a rate model. So that's what I'll be showing here. Within the same, we said that the excitation in this network comes from neurons on the same side of the integrator. So we characterize those by a weight matrix Wij. And we said that the inputs go through some potential nonlinearity, S excitatory at the excitatory synapse. Or for the inhibition, we said that there's also a strength of connections Wij, but these go through potentially a different nonlinear S nonlinearity, S inhibitory, driving inhibition in the network. There are also in this system what are called tonic background activity. That is just neurons who are continually firing and providing background input to the system. It turns out that when the head moves, those tonic background inputs no longer are tonic and they actually vary. But for what I'm showing here, the head is fixed, so those are constant. And then there's the saccadic burst commands that I showed you earlier. These are the brief velocity encoding pulses that move the eyes from one place to another. Now, this is clearly, and we also have a threshold that neurons can't fire below 0 firing rate. So now this is the full glory of the nonlinear system, but one can quickly ascertain a very simple condition to make this an integrator. And that is if all of these terms here, basically the intrinsic leak, the intrinsic decay processes in the neurons are offset by by the feedback coming from the external inputs. If these all sum to 0, then the derivative of the firing rate is proportional to the burst command, or the firing rates are integrators of the burst command inputs. And we return to the simple idea of feedback offsetting decay processes lead to integration in this system. So I'm not going to get into it today. You can just write down this balance condition, just as actually in the way we formulate it as a simple regression problem, and therefore tune the connections. The bigger challenge is not how to make it work. The bigger challenge is actually how to figure out what of the many degenerate possibilities actually are the ones that might correspond to experiments. You can ask me about that later, but I'm not going to get into that today. Here, I'm just going to sort of quickly wrap up this part of the story and say that one can do this. One can actually do it in a full biophysical spiking model, and that's shown here. So there's actually showing the spiking voltage of one of the neurons in the biophysical model to a saccadic input. The neuron started out silent. A brief saccadic command came in, a velocity encoding command came in. You see the neuron responding to that directly, but then after the command turns off, what I want to draw your attention to is this persistent activity. Another command comes in and that's integrated, and now after that command turns off, there's a higher rate of persistent activity, and so on and so forth. This is showing a summary, actually just showing the firing rate of one neuron, but now just smoothing out these spikes into a slightly smooth time average firing rate. Green just is to guide your eye of a perfect integral, and gray is actually the raw smooth firing rate, and black is with a little bit more smoothing. And you can see that this neuron is perfectly integrating its inputs. And in fact, the model, I showed you before that there are many different neurons, each of which have slightly different thresholds and slightly different slopes. And you can see those slopes and thresholds are taken from the experimental data. That's actually the solid line. The box plots are actually from the neurons in the model with the noise associated with them. And what you see is basically that the model can recapitulate the activity of each of the neurons in the experimental system. And this at the time, I think, was one of the first studies that really could recapitulate the activity of across a network at this level of precision, and certainly for a memory network. All right, so that is, in the spirit of how these talks have gone, that is the end of part 1. So I could take a break for questions. So that was really to illustrate the positive feedback mechanism and connect it to a real experimental system. In the next 2 parts of the talk, I will go on to sort of question whether every system uses this positive feedback mechanism and what some other possibilities are, but I'll break for questions. And if there are no questions, I'll go on. Bill, I guess you can moderate the, if people have questions in chat, but otherwise people can feel free to shout.
So in addition to the breaks that Mark has scheduled in, you're welcome to put questions in the chat, and if it seems urgent enough, I'll interrupt. Particularly good to hear from students for whom this may not all be familiar. So
may I ask a question? Yeah. Can you hear me?
I can hear you. Great.
So my question is this. So I understand that like when there are these excitation stimuli, it excites, right? Then for the stimuli to decrease, you need de-excitation stimuli, like which sort of decreases the area under the curve in the integrator, right? Mm-hmm. But suppose in real systems, there is no de-excitation stimulus, but the stimulus, can it come down by normal decay, like a dissipation kind of a decay? Is that
right? So so the main idea is that sort of neurons in isolation in these models, they all decay. So the natural tendency of the system would be to decay back to some baseline. If in this case, the baseline in this system is not 0, but determined by those tonic background inputs. So it decays to some constant activity level. And then it's the recurrent connectivity that actually allows the network to maintain activity at an elevated level, or actually also maintain activity at a level below the background activity. So it's really, the short answer to your question is yes, there is intrinsic decay, and if things aren't tuned properly, which I will talk about next, then in fact, if the feedback is not strong enough, then all you will see is activity decaying away.
I see. Okay. Okay. Thank you.
Unmute myself. Mitch, why don't you had a question about adaptation mechanisms?
Yes, thank you, Mark. Yes, I was wondering whether in what you've talked about now or what you will be talking about in the remainder of the talk, how intrinsic adaptation mechanisms in terms of excitability may figure into this, what roles they may play. So two things that probably do similar types of things are things like paired pulse depression, for example, at the synaptic end, and then spike frequency adaptation at the intrinsic end. But both of them are dampening mechanisms to a fixed level of input activity. And what role do you see that in these kind of memory processes, or is that something that you're going to go into?
I'm not, so I'm glad you asked.
Okay.
So it's a good question, and there's a long history of research on this. Misha Tzadok's, who's now at the Institute, at least part of the year when he's not in Israel, has thought a lot about the particular one of thinking about synaptic dynamics. So in this system, well, just from the modeling point of view, adaptation is usually sort of the curse of these types of models, and in some sense, and it tends to be destabilizing. So there is a very rapid spike rate adaptation experimentally, but it's on the more tens of milliseconds time scale and not on these long time scales of many seconds. If lined up correctly, and it will get into the next talk, it actually could be helpful if you can think of adaptation as being like a derivative-like operation. So it's sort of the opposite of integration. If you have a system and there's an input and then you jump up the input, then an adapting system will jump up transiently and then come back down, and that's a derivative operation. And that's the next where I'm gonna go next is where derivatives could help you. So yes, I should have said I will be picking up on that, but generically it's the opposite of what you want to do. I mean, generally, when you don't do anything except put those processes into these positive feedback architectures, what one gets is a destabilization, because they tend to respond to really strong fluctuations and they tend to not hold on to the persistent activity. So usually when it comes to things like synaptic dynamics, what people have instead looked at is things like synaptic facilitation, which would be paired pulse facilitation, where when you have an input, it now biophysically brings calcium into the terminal and makes it more likely that there's transmission at future times. And that provides an intrinsic mechanism of memory, which can lessen the demands on having such a precise network architecture for tuning these weight matrices.
Just as a follow-up, what if an adaptive mechanism, a negative adaptation mechanism that we're just talking about, can be tuned down in a rapid way, so that that what certain patterns of inputs might do is actually suppress adaptation mechanisms.
Right.
Is that something similar to what you're talking about in terms of facilitation, that might have an ability to help sustain memory traces of sorts?
Yeah, there's a little bit of a chicken and an egg problem here of, the first thing you would do if you were engineering the system is not have the adaptation mechanism at all.
Right.
But if you had it, that might then call for some other mechanism that suppresses it.
Okay.
What I should say is that a remarkable feature throughout the oculomotor system is that neurons can maintain for long periods of time hundreds of spike per second firing rates, which is very, very high. So it seems like this system has actively worked to suppress some of those mechanisms. And in fact, there was some work from Sasha Delac who sort of found that it's not that the neurons are just like a theorist neuron. It's more that basically there's adaptation and then something else kicks in and offsets that. And then that new thing that kicks in starts saturating perhaps, and then another process kicks in. So I think the neuronal biophysics handles a lot of that, and that's something we haven't really delved into in depth, except to see that at the end of the day, the neurons are remarkably linear in their properties.
Thank you.
Yeah.
So Mark, we have a very general methodological question, and maybe if I can amplify it a little bit, if you could go back just a little. Okay, so let's stop here. So, I mean, the question is about, what methods are you using? So maybe you could say something, you sort of amplify a little more. How do I go from writing these equations to the figures that appear on the next slide?
Yep.
For some of the students in the audience.
Right. Okay, so that's a good question, because like I said, at this time I did brush under the rug a lot. So what we did methodologically, let's first just pretend this intrinsic, let me just think how I want to say it. So what we did, let's just say what exactly we did. So what we really did here is the opposite of what you do in the differential equations class. In differential equations, I give you this nonlinear differential equation and you say, I'm hopeless because it's a terrible nonlinear differential equation. But here, we actually turned it into a fitting problem. We said we know the firing rates experimentally. And we had enough neurons that were recorded that we put them together, and we pretended that they were all recorded together, even though they weren't, because statistically they seem to do the same thing from animal to animal and neuron to, and, you know, and from time to time. So we put in the firing rates. And now once we put in the firing rates, and in this F, we actually just injected current to see how a neuron would respond to current injection, and calculated, basically fit a function F there. And now I said for persistent activity, these must sum to 0. So now we just have an equation, F, which is known, of firing rate, which is known, equals this big sum here, sum Wij S excitatory of R plus or minus Wij S inhibitory of R plus a tonic background. So then what we said in this case, one can do more complicated things, as we said, let's just assume we know what this function S is. If we know what this function S is, and we know what those rates are, we just turn this into a linear regression problem. We just fit the function F of R is Y, and that equals sum of Wij X plus a constant. So that's literally what we did. If you want to say sort of in detail what did it mean, because I think you can fit things without actually thinking about it, what it really said is, as the firing rates go up, this most simply would be a term minus R. The leakage, as neurons' firing rates go up, they tend to get pulled down harder. Well, on this side of the equation, as the firing rates go up, we tend to get more excitation, and we tend to get less inhibition. And that's offsetting that intrinsic leakage. And what you're really doing is you're basically just trying to line up two functions, this intrinsic leak, with a sum of basis functions, really. So if we think of each of the other neurons, Rj, after being put through these functions S as just basis functions, we're really saying, can we sum up with weights that we get to choose as modelers, Wij, a bunch of basis functions S of R, and make them equal this function F of R taken from experiments. So that's how we actually fit them.
Good. And then we have one more question in the chat. Is it roughly understood why goldfish use 100 neurons to encode the 1 or 2 dimensional orientation of the eye? Is the network size related to how slow the slowest node can get?
Yep.
Actually,
so
there's 2 questions there, I guess. But
yeah, so it's a good question. So why? So this is one of the things. So now I'm working in collaboration with Emory Oxide. We've been looking at the larval zebrafish, where we can do a lot more. It turns out, and I'm not getting into it today, but it looks like the activity is a little higher dimensional than this, and there's a few reasons for that. It may be that the eye has a few more time constants in it to control than just the simple models of the eye being a spring in a viscous dashpot. So that then calls for having a couple more time scales, but even then it's probably still less than 10. So we have, an order of magnitude more neurons than number of time scales that we need to generate in the system. And another possibility is there's slight differences in different behaviors. So I'm talking about fixation behavior, where a saccade comes in, but actually the dynamics of tracking eye movements, where if I move my hand back and forth and ask you to track it, that comes through a different set of inputs. And it looks like the system is capable of actually maintaining independent representations of those. So maybe that's another factor of 2 for dimensions, but we're still about an order of magnitude off. So turns out when you do this fitting problem, in general, now you're not fitting linear, you're not doing linear networks where you just have to get an eigenvalue exactly equal to 1 in linear network theory. What I said is you're really doing a basis function approximation, where these terms S excitatory of R and S inhibitory of R are being added up with different weights to offset this function F of R. And in general, what you really have is a sequence of very tightly spaced fixed points. And the more basis functions you have, which is to say the more neurons you have, the more tightly you can space those fixed points. So in terms of the precision, actually having more neurons gets you more precision. It doesn't necessarily get you more robustness, although that depends on how it's laid out. If the system is distributed sort of spatially, then you could have more robustness because maybe the animal gets a stroke in one location and now locations far away from that are safe. But those are sort of those are the basic phenomena, that you get tighter spacing of fixed points, and that potentially you're more robust if the large number of neurons are laid out in a way that is useful for conveying the robustness. And that's a question I'll talk about next. I won't talk about how they're laid out, but I'll talk about the general idea of robustness.
Okay, great. Let you keep going. I will.
Okay. So two questions I want to ask for the next parts of the talk are first, I just gave you the dogma, which is positive feedback loops are what maintain activity in a system that naturally has decay processes. The positive feedback offsets the decay processes. Is that the only way integration can be performed? And then could alternative mechanisms describe short-term memory activity data? And in particular, does all of the data look like the data I just showed you from the fish? Is the fish a good model system for other neural integrators and for other short-term memory networks? So that'll be the next two parts. So the first, just the introduction to all of these, is that what I said was that the positive feedback mechanism forms a line attractor. And the issue with a line attractor, and this relates to the question that was asked, is if there are divots in that line attractor, then you're going to get discrete fixed points. Even worse, if that line attractor is tilted, you're just going to get a systematic drift. And if that line attractor, let's say, is a bowl, everything's going to decay back to a single fixed point, which is what happens when the feedback is not strong enough. But the main point is a line attractor, geometrically, it's a fragile structure. So if the surface isn't flat, the network activity state is going to slip. And if you really want to have analog memory, it would have to be perfectly flat. So what are the hypotheses for how the system can be robust? And one that I won't talk about and something we work on is plasticity. So the thought is that plasticity in the system I showed you, probably mediated by the cerebellum, is continually reshaping the trough to make it flat. So that is likely going on, but from the experiments that started in David's lab and have continued in Emory Oxai's lab, that's probably on the tens of minutes time scale. So what keeps things robust on a faster time scale than that? And a couple hypotheses that we've been working on are, well, maybe it isn't really a perfect integrator. In some sense, it's never a perfect line attractor. As I pointed out, once you have a nonlinear system, you're always making discrete fixed points, but maybe you can make those discrete fixed points particularly robust. And if I have time at the end, I'll talk about those. There's also some beautiful work from Alex Kulikov and John Lissman that shortly preceded ours. And so I encourage you to look at that. The one I want to talk about immediately is maybe there's a way to effectively have this attractor landscape filled with viscous fluid. In other words, can we add molasses to it? And here's what I mean. Here's what I mean by this. And it basically comes from control theory. And this is going to be now getting to Mitchell's question about negative feedback. So there's a basic principle of control, and that it's that if you have a system where you have an input X and an output Y, if you have very strong negative feedback, and what I mean is that the feedback is strong enough that in some sense it dominates whatever the intrinsic system transfer function G is, then the output will equal the inverse of the negative feedback signal. So if I were to feed back with negative feedback, that's the minus sign here, a derivative of the output, then the theory says you will get that Y is an integral of its input. And engineers really, I think would think, why are you building a system with positive feedback? Positive feedback tends to run away. Can't you build the system with negative feedback? Well, the right negative feedback for this system is not to negative feedback to a single fixed point, but the negative feedback to a continuum and make an integrator. So the feedback needs to be not slipping. And that's a derivative, because slipping is a change or a derivative. So that's shown here. And I should say this is work with my former postdoc, now at NYU Shanghai, Sukbin Lim. So here's the basic toy picture. Suppose we have a memory cell or a memory population, and we just say we're going to build a slip detector. Whenever it starts slipping, it picks up on that, takes that slip, calculates minus d by dt, and feeds that back with negative feedback. So when we now took our system and we basically add to the right-hand side W derivative, that's the strength of the feedback, dr dt. And what I want to, and clearly just mathematically what this does is W derivative acts as a long time constant. You could just put it on the other side and make the new time constant in the system tau plus W derivative. Physically, of course, this is like viscous drag. You know, it's proportional to the velocity. So in that sense, this is why we have this picture of molasses, that we basically added viscous drag to whatever system we had before. And this is, I should say, this is complementary to the positive feedback picture. You can always take whatever system and add this mechanism on top of it. And the point is that there will be a corrective feedback. If the firing rate starts drifting upwards, this will tend to push it down. If the firing rate is drifting negatively down, this will push it up. And of course, this alone won't make a perfect fixed point at any point. I mean, it's still molasses, it's still drifting down, but the point is long time scales could be generated. Okay, so how do you do that within a neural network? Where could these derivatives come from? And there's many mechanisms, and you know, Mitchell, this is where, you know, one thing you could do is have a cellular mechanism which is derivative like spike rate adaptation or synaptic depression, and use that. I'm going to focus on a network mechanism here. And the simple network mechanism just takes advantage of writing out what a derivative is. So negative of a derivative is r of t minus dt minus r of t. So this says if I have my population whose firing rate is r of t, what I need to have negative derivative feedback is to have fast negative feedback, that's the minus r of t, and then slow coming in at a delay, positive feedback, r of t minus dt. And so negative derivative feedback arises when positive feedback is slower than negative feedback. And then the other thing is, notice it's implicit that the coefficients of these two terms are 1. So there's a balance of excitation and inhibition. And one of the intriguing things for those who know neuroscience and know things about cortical networks is in many neocortical networks, people have found that there's a balance of excitation and inhibition. So we think this might be a key component of neocortical mechanisms of persistent activity. More to flesh this out and not make it such a cartoon, more in a little bit more biophysical sense, we think of two populations, an excitatory population of neurons and an inhibitory population of neurons. And now the slow positive feedback is this excitatory to excitatory loop. And then the negative feedback is the excitatory loop coming back through the inhibitory neuron to provide negative feedback. And I should just note that in frontal cortical networks, people have found that these excitatory to excitatory connections have relatively enriched NMDA receptor content, and that's a slower receptor. So it looks like this connection might indeed be slower. And in fact, the excitatory to excitatory connections are slower than the excitatory to inhibitory connections. So it seems like the timescales at least line up in the experiments. And again, people have found that excitation and inhibition, when people record and actually look at the excitation and inhibition coming into cells, it looks like those have some degree of balance. Or, you know, and appear to be, in how tight that is, I think is, you know, still an issue being worked out, but at least the general theme of cortical networks is an excitatory inhibitory balance. And I'll say it doesn't need in this work to be, I'm not going to get into it, doesn't need to be super tightly balanced. What you can think of all the things I'm doing here is really a mean field theory calculation. There's other beautiful work that I encourage you to look at from Sophie Deneve, where there's really a true perfect fine-tuned balance of excitation and inhibition based on a predictive coding framework of control theory. Okay, so let's just take this idea of having, you know, balanced excitation and inhibition and positive feedback slower than negative feedback to make negative derivative feedback. Well, if we put it into our network from that architecture, if you put in pulse inputs, then what one finds is the firing rates indeed look like they respond and have long, slow time scales of decay. This sorry, I lost my time scale, but this is the time axis, and this is on the time scale of seconds. And if one puts step inputs in, one sees one is getting an integral. So a step is being integrated to a ramp. Now, the interesting thing about this, which gets to the issue of robustness, is that these systems appear to be more robust to many of the typical types of perturbations we think about for neural systems. And I should say they are not robust in general. There is a fine-tuning condition in these. It is that balance of inhibition and excitation. But given that fine-tuning, there is robustness against many common perturbations. So here I want to show just my character of the positive feedback networks that I showed you in the previous part of the talk, with just, a population with excitatory feedback onto itself. And the issue here is, if you, for example, killed a set of those neurons, if the set of them were silenced, or if all of the weights went up in their strength due to maybe a global neuromodulatory input, even very small increases or decreases in the intrinsic gains of neurons, cells, excitatory or inhibitory cells being killed, scaling of synaptic weights will lead to dramatic, either if it's increases, dramatic exponential growth in activity or dramatic exponential decay in activity. Now I want to look at the negative derivative feedback network. So in this one, imagine that we were to silence a fraction of the excitatory neurons. Well, we haven't changed the time scales of having the positive feedback still be slower than the negative feedback. And furthermore, we haven't changed the balance of excitation and inhibition, because when we remove a fraction of the excitatory neurons, we're decreasing the excitatory input by that fraction, but we're also decreasing the negative feedback loop by the same fraction. So even though this is a system that is more like molasses and there's a slow decay, here even 10% increases or decreases in numbers of neurons or gains of neurons leads to barely any change in the decay. And of course, these two mechanisms are not mutually exclusive. So here what I've done is I've added a little bit of positive feedback to flatten out, if you wish, the attractor surface. And then again, 10% increases or decreases in activity allow the activity to be persistently maintained with very little effect of these perturbations. So we think this is an interesting mechanism that has certain robustness properties to it, as well as, perhaps being much more characteristic of what we think of as neocortical network structures, of having this component of negative feedback as well as positive feedback that together can work to provide a negative derivative feedback component. All right, so that is the end of this mini section of the talk, if there's questions on that.
Do we know anything experimentally about robustness? I mean, people always say it's a good idea, but and it feels like it, but what?
So one of there was a really cool experiment that was done by Amy Arnston. I think she's at Yale, and what she did is she did two different manipulations. One, she manipulated, I believe, I think she inactivated some set of neurons and memory was very robustly maintained. But then she inactivated a particular subunit called the NR2B subunit of the NMDA channel. And the neat thing about that one is it's only in the excitatory to excitatory connections, or it's predominantly, I should say, in the excitatory to excitatory connections, but not in the excitatory to inhibitory connections. And then the memory failed miserably and was terribly not robust. So that's the most suggestive, experiment at that level. I think within the goldfish, it's been a puzzle that we know that after doing many experiments, many cells are being killed. I mean, a glass electrode is going through an area which only has order 100 neurons, yet the system seems to maintain its behavior quite well. So that was one of the initial puzzles in that system. And we still, I'd say, don't know exactly what the answer is there. On longer time scales, the cerebellum could be retuning the network. On shorter time scales, if I have a chance at the end, I'll talk about some possible intrinsic mechanisms that might maintain the activity. And the other thing is, I said there may be 100 neurons. The connectomic work from Sebastian suggests that there may be, the integrator area may have been much more extensive than we thought. So we may not have been taking out as much positive feedback as we did, and then the standard theory may actually still hold.
Okay, thanks. If there's nothing else burning, maybe you should go on, and that'll leave some time at the end for more general questions.
Yeah, and then there'll be part 3, and there's one that there's sort of a part 4 if there's time, which relates to some of the things that Gordon talked about, and it's only about 5 slides if we have time. But let's see about this one first, because this next section's a little bit. So here's another experiment, and again, this is a neocortical recording, and this is something that has come up. Was at the time I found it was very unusual in the literature, but has become more and more commonly seen. And this is 5 neurons recorded in frontal cortex during a memory task. This was actually recorded by a person at St. Petersburg University, Batuev, and published this in 1979. And what he found was that he found neurons so what the task was, I won't get into the details, except that it was a task where information needed to be maintained over about a 9-second delay period. And he found some neurons that maintained activity during the early stages, but then their activity decayed. Others that maintained activity during the middle stages of the task. Others that came on at the late stages of the task. Some which look like sums of early, mid, and late stage neurons, but only 5% of the neurons look like the classic persistent activity neurons that people had been talking about in the literature. Whereas, here, these numbers are more in the 15 to 20% category. And that really does not look like our classic attractor models. And here's sort of the reason why. I think it should be fairly self-evident, but in the traditional attractor networks, the activity is low dimensional. That means all the neurons go into the same, after the initial transients die away, go into the same pattern of activity. So an input comes in, and after some transients wear off, all the neurons in this attractor decay together. And it's due to this, and ultimately it's due to the strong coupling that mediates the positive feedback. So we made us think, could we come up with a completely different network that would still solve the task, but doesn't have the attractor structure? And the simplest, stupidest one we could come up with was a chain of neurons, or a chain of groups of neurons. So we make groups of neurons that each are characterized by just linear dynamics with a filtering time constant tau. And then we chain those together. So we put an input in, and this is showing you the first one would have just been an exponential decay. Here's the second one in the chain. Here's the 5th, the 8th, and so on. And it's just progressively filtering what was filtered before it. And what's interesting is if one just takes all of these, it's a 2-line proof. If one sums these up, if it's an infinite series, one actually gets a perfect integral. This is a linear system. A delta function of unit amplitude turns into a step of unit amplitude. And that's being shown here. This is just due to finite size effects that it's decaying away here. And it's when the weights all equal 1 in the network and in the readout. Here's where I allowed the weights to be a little bit larger, and you can see that it's maintaining activity over a longer time scale. And in general, in terms of the scaling, not surprisingly, activity can be maintained over a time scale proportional to the length of the chain and the time constant of the elements in the chain. Okay, so this is just showing it was a linear model, so it has to work. But if you put steps into the network as the input, shown here, then you get beautiful ramps of activity. And again, here is where it runs out the end of the chain. So there's a limited duration over which it can respond. And here's just fitting that data. So here's just trying to fit with an attractor network where all the neurons decay to a low dimensional structure. And of course, one can't possibly fit neurons like this where there are early and middle and late bumps in activity. But with the feed-forward network, what the network has really done is formed a basis. It's formed a temporal basis where each neuron represents a certain point in time. And from that basis, it's a much richer representation than just maintaining persistent activity. It can maintain much, many different patterns of activity. And I think, maybe much more useful as a general computational backbone for many different computations. So, and this is just showing one of the first studies that actually found cells like this. This is from Chris McDonald, who was in Howard Eichenbaum's lab. And this was a task that I should say was not a spatial task. It was just a memory task. And something needed to be, a item needed to be remembered starting at time 0. And here's this feed forward progression. And I should say, sorry, each line of this is a neuronal recording. So again, you see these neurons at the top fired early, these neurons later on fired at later times and more spread out. And it certainly, at least qualitatively, looks like it resembles just a simple feed forward chain effectively. Of course, I'm sure, not linear, but the effective architecture looks like a chain of activity. So we wanted to push this a little bit and say, okay, so a feed forward chain seems too simple, and it would have to be something effective from a network. But can we generalize this to more general architectures where there might be these feed forward dynamics, but they might not stand out so obviously and hit you in the face? And here was the idea. If we have a feed forward network, we have the red neuron goes to the blue neuron, goes to the purple neuron. We make a connectivity matrix that we can see is just lower triangular. And geometrically, we can think of, instead of thinking of neuron 1, we can think of the state 1, 0, 0, transitioning to the state 0, 1, 0, transitioning to the state 0, 0, 1. Well, mathematically, we can certainly just take that weight matrix and apply a rotation to it. So what if we apply a rotation matrix? Then what we're going to do is we're going to rotate this picture where now some more general pattern of activity goes to some orthogonal next pattern of activity, goes to some orthogonal next pattern of activity. But the actual architecture will be a very complicated, recurrently connected network with excitatory and inhibitory connections, and something that maybe could be going on in something intricate like a neocortical network. The mathematics behind this, I should just make a note, come from the Schur decomposition, and there's a lot of beautiful linear algebra sort of underneath this sort of simple idea. So, here we go, just showing that this works. So if we put input to the first neuron of the feed forward network, the first neuron decays exponentially. The next neuron goes as t e to the minus t. The next goes as t squared e to the minus t. And same thing occurs if we first, in this rotated functionally feed forward network, if we stimulate the first activity pattern, then that activity pattern decays away. That is the set of activity, its amplitude will go down. It then hands off to the next activity pattern and to the next activity pattern. But that might not be so obvious if you look at the neuronal firing rates, because the neuronal firing rates are just going up and down and wiggling. And while you may see a little bit of a transition, some earlier and later neurons, it's not at all obvious sort of what's going on if you just looked at the firing rates. And finally, I should say the same property occurs that if you sum up those firing rates, you can get a perfect integral of the input and perfect persistent activity.
And Mark, just to be sure I haven't missed anything, the statement that the weighted sum isn't changing there is a statement that if I look at the equations right there, there's a global conservation law, which means that in terms of the synaptic weights, there's been some fine tuning, right?
Yeah, so this is definitely fine tuned. I think I have it in here.
Yeah. I think you said that. I want to be sure I didn't miss something.
Yeah.
Right, because generically, if I pass things along a chain, it decays away, right?
Yeah. So generically, it could do either. So really, especially I feel like now when everyone's doing recurrent neural networks and doing back propagation through time by unrolling their networks, that's really what we've done here. So the feed forward chain, in some sense, is just unrolling the recurrent network in time. So now you've just taken, instead of the neuron at time step 1 hands off to the same neuron at time step 2, now it just hands off to the next neuron. And that immediately says we're going to have the same fine tuning problem because the handoff needs to have weight 1. Now, there are some benefits that you could start growing a little bit and then have some later connections that start decreasing. So there's a little more flexibility with those extra degrees of freedom. Running out of the end of the chain means that you won't have catastrophic growth because eventually the activity needs to decay. And we and Surya Ganguly have pointed out that that could help in terms of not building up noise in the network when there's sort of a constant background of noise ongoing in the system. But the same general principle definitely holds. You still, if you hand off a little bit too strong, and then that neuron hands off a little too strong to the next one, and that one hands off a little too strong to the next one, you will get exponential growth, and vice versa. If it's not strong enough, you'll get exponential decay.
Is it true that is there something where you can have the individual things fluctuate, but somehow you think each you need a 1 at every stage, but can you have a 1 plus epsilon? Can you have some number of 1 plus epsilons and some number of 1 minus epsilons and all?
Yeah, so you can, and indeed, because and I think we'll see where the math sits on that at the very end. There's some cute math which comes out of what's called pseudospectral analysis. But yeah, so that is something nice, that there can be a compensation. It can start growing and then decaying. Now, if you wanted a perfect integrator, then you're going to start messing up your perfect integrator because it may integrate too much in the beginning and then too little in the in the intermediate period. But yes, you can start growing and then have the connections not be so strong and have it bring it back down so it jiggles back and forth.
Thanks.
Yep. Okay, so here's the puzzle, and the puzzle that I was just alluding to. So line attractor networks, what I referred to earlier just with the linear theory, is the classic line attractor has an eigenvalue of 1 in the weight matrix. This is just linear networks. So we have that 1, I'm not showing 0. That 1 is in the weight matrix because I'm saying that there's a decay term, an identity decay term of value -1. So the overall system would have an eigenvalue of 0, and this is just on the weight matrix. And then, most simply in the classic line attractor, all the other eigenvalues are less than that in their real parts, so that we end up with rapid, some modes which rapidly decay and then persistent activity. Here's the math puzzle. Feed-forward networks are described by a lower triangular matrix. When we know that for a lower triangular matrix, the eigenvalues are given by the values along the diagonal, and those are all 0. So where did our persistent mode go? Clearly in the behavior, we're seeing that activity can persist for times of order n times tau, but in our eigenspectrum, that seems to have disappeared. And the right way to solve that is with something called pseudospectral analysis, which there's a beautiful book if you're interested in this. It's sort of the Bible for this by Lloyd Trefethen and Mark Embry called Spectra and Pseudospectra. But here's the sort of brief summary. So eigenvalues that we conventionally use in network analysis satisfy, of course, the equation W minus lambda the identity times V equals 0, or WV equals lambda V. And they govern the long-time asymptotic behavior of the system. And indeed, in this system, which is a feed-forward chain, the only long-time persistent behavior at every point in time, activity shifting from one location in the chain to another. So it's not an eigenvector. The only thing that is an eigenvector is the very last neuron. The very last neuron, if you inject current into that, it continues to be its activity decays away with the intrinsic time constant of the neuron. And it's the same pattern. It's just that last neuron firing. So indeed, the eigenvalues do describe the long time asymptotic behavior that at the very longest times, the only thing that's active is the last neuron. Pseudo eigenvalues are the set of all values lambda epsilon that satisfy an inequality that corresponds to almost, if you wish, being an eigenvalue. So now they satisfy W minus lambda pseudo eigenvalue times V is not 0, but is less than epsilon, so that it's almost an eigenvalue. And therefore, there can almost be a perfectly long, there can be long time scale modes for very, very long times. So they govern transient responses. And they can differ greatly from the eigenvalue when the eigenvectors are highly non-orthogonal. That is non-normal matrices. And coming from physics, as I and this audience did, we're used to dealing with Hermitian matrices. We're used to dealing with orthogonal eigenvectors. So this is really a property of being non-Hermitian, about really being non-normal, non-orthogonal. And of course, this is a strongly asymmetric matrix when you have a feed-forward network. So here I'm going to show you, again, here was the line attractor. But if you want to think about the feed-forward network with just one little tiny perturbation to it could be a cyclic orthogonal network. If we just took the feed-forward network and took the last neuron and connected it back to the first one, that's a normal matrix. All the eigenvectors are orthogonal, and what you get for a spectrum is actually a circle of eigenvalues, and you get activity patterns that look identical to the feed-forward matrix until such time as the activity cycles all the way back to the first neuron. So the feed-forward networks, when you calculate their pseudospectra, show exactly this behavior. The pseudo-eigenvalues form a circle, and that circle is within epsilon of having an eigenvalue of 1. And in fact, it has many eigenvalues that are very long. And in the theme of thinking about generating multiple time scales, it's really, you can think of this as generating many time scales over prolonged periods and a diversity of them. All right, so that is my, that's the end of that section. And I just want to summarize this, and then if we have time, we'll do one other little section that may relate to things that Gordon talked about in the last talk. But I'd like to summarize here. So we said that short-term memory on the 1s to 10s of seconds time scale is maintained by neural activity patterns that persist following the offset of a remembered stimulus, whether individual neurons or the pattern of sequentially activated neurons. Possible mechanisms are tuned positive feedback, which is the essence of low-dimensional attractor models. Negative derivative feedback, which opposes slip, so it's velocity opposing. Or in the last section, feed-forward chains, possibly in disguise due to a rotation. And I, again, want to point out for this that the dynamics in this case is very much not low-dimensional. It's high-dimensional dynamics, and therefore it can produce many different temporal response patterns across many different timescales. If people want me to go on, 2 other things I could talk about, and I think the last one probably there isn't time, but the other one may be more fundamental and there is more time, is where bistable components, or really having sort of digital elements within a network, can really save the day when it comes to this robustness problem. But I'll leave first to questions on the last section on the feed-forward integrator.
Yeah, may I ask a question?
Sure.
Yeah, can you speak a bit more about that pseudospectral? It's like when is this relevant? Because it looks like really interesting. This, yeah. So I mean, is it like that the origin, the eigenvalues are relevant when we are looking at like long-term dynamics, but when you're looking at short-term dynamics or short-term responses, the ideal way to do this is through the pseudospectral analysis and not through the spectral analysis? Is that the philosophy? Or?
Yes. So, I mean, what I've shown you is there's some very long time scale dynamics here, which were not captured by the eigenvalues. So the eigenvalues capture in the limit. As t goes to infinity, the eigenvalues will capture that. And in the chain case, that's when activity is only present in the last neuron in the chain. What was surprising here is that one could get, even though, so in general, you think of pseudo eigenvalues as describing transients. But the issue is those transients can last a really long time. So I would instead answer the cautionary tale is, if you have strong asymmetry in your system, in your matrix, or more mathematically, if your matrix is strongly non-normal, that's where the pseudospectra start becoming relevant. And there are several different ways one can define non-normality. You can look those up, but that's the signature. If you see something that is strongly non-normal, be careful with your interpreting your eigenvalues and eigenvectors. And this case was an extreme. This one was truly degenerate, and there was only a single eigenvector. And I could have added epsilon perturbations to that to make it have a full set of eigenvectors and a full set of eigenvalues, but you still would have ended up with something sort of confusing if you thought of it from the point of view of eigenvalues and eigenvectors in terms of interpretation. So, but the math would have worked out fine.
I see. Okay, so it's about non-normal matrices for which the pseudospectral analysis is relevant. Like, is the situation where there is a huge degeneracy in the eigenvalues a case where this should be relevant?
Yeah. So the degeneracy, you can have, the degeneracy was one issue, but that really should be separated, because you can have systems which are degenerate, but where they are largely, where the dynamics are largely normal and all your timescales are fine. And sure, you have two eigenvalues which are degenerate, so you can't identify as, so then you are only defining the space span by those two eigenvectors. But this is really about the non-normality.
Okay. And one more question about that molasses thing that you said. So the role of adding the molasses is I think I mean the way I was thinking about it is like, is the role of the molasses to increase the time scale of the processes occurring? Like, is it just like it slows down the process, slows down the dynamics? Is that the role of the molasses?
Because the role, sorry, I missed. Is that the role of what?
The friction term in the previous case, in the section.
Oh, in the previous case, so the friction term, the role of it is to oppose slip and therefore slow down the dynamics. And you can see in the sort of toy model I made that it literally is equivalent to a time constant. The neat thing is, because it's made from a network phenomenon, you can dial that up and down. So it's a more flexible than if you just had a long time scale at the intrinsic neuronal level.
Okay, so is it like that the more and more we increase the friction term, it becomes more and more robust, because then the dynamics will become more and more slow, the decays will become more and more slow? Is that true?
So the term robust is a tricky one. So it becomes slower and slower as you turn up the friction, because it's like making your viscosity thicker, larger and larger.
Exactly.
The robustness in that network was made by a balance of inhibition and excitation. And that is still a fine-tuned condition. And so you're no more robust against that.
Okay, I see. Thank you.
May I ask a question?
Sure.
Yeah, this was, so, and again, my questions, as you can probably tell, they come more from the kind of electrophys and not from the mathematical understanding. But one important thing about brain networks is that they're very excuse me, I'm sorry, I have to.
Is there another question? Well,
I'm sorry, I'm back.
Okay.
I'm sorry. My question was, the brain is very sensitive to a whole host of genetic alterations that that in some ways seem conceptually distinct from each other, but can all lead to sporadic seizure susceptibility. And in some way, intuitively, it seems to me that to get the robustness and the precision in your system, there are so many parameters that need to be fine-tuned, as you say, that I'm wondering to what degree your network models basically are in some way predictive of the sensitivities of networks to seizures in a variety of genetic or environmental insults. It's kind of a wild question, but I'm wondering whether, in other words, you can simulate seizure susceptibility in your
So
models?
yeah, so I even sometimes say when I give talks about the positive feedback networks that indeed, I sometimes show what happens if you decrease the weights, activity decays away. What happens if you increase the weights? The network goes epileptic.
Mhm.
That's exactly what happens. So I do think that there is probably strong positive feedback. A lot of it is in, a lot of seizures, for example, originate in the temporal lobe or around the hippocampal circuits, where we think of storing memories through positive feedback type mechanisms, and they're very susceptible to blowing up in this way due to positive feedback runner I. So I think that is the basic idea, and that positive feedback can come from many different sources, whether it's a genetic mutant of a sodium channel or or a chloride channel or something else.
Can you see in any perturbations you make to your network that can result in sporadic seizure? Because I know obviously if you substantially change the behavior to have more excitation overall, the whole thing explodes in terms of becoming. But the more realistic issue of seizure is something that the brain is functioning normally 99% of the time, and it goes off the rails sporadically. Can you see any of that kind of behavior in your systems?
So the simple one, which leads to seizure-like activity, is actually blocking the spike frequency adaptation channels. So that fits. I say that because of your last question. And then you see exponential growth in activity. You can also tune the network so that you put it in a virtual world where having exponentially growing activity actually tracks the world, because you set up the world that the world moves in an exponentially growing way as you move your fed back from your eyes. And those are some beautiful experiments done by Guy Major and David Tank's lab. And that seems to be controlled by the cerebellum, and various cerebellar disorders lead to sort of very strange, intricate dynamics where things go haywire periodically. And that's likely because the cerebellum is serving as an extra feedback loop that can end up being in a positive feedback form. And there's various genetic disorders that will lead to all sorts of interesting forms of nystagmus, which is with the eyes moving in non-fixed, non-stable fashion.
Thanks.
Yeah. The next part of the talk, if I go there, is about what you might do to make things more robust, fundamentally robust against these issues, which I said is only a few slides.
So Mark, why don't you try and fit in these few slides and this is where we are.
Okay, so I will do this pretty quickly. So here's just a cartoon of what I've said before in the linear analysis. Just imagine a neuron connected to itself with a weight W. You have a decay term -R and a feedback term WR. Again, this, if W is greater than 1, is an exponential growth equation. If W is less than 1, is an exponential decay equation. That's being shown here. I'm plotting as a function of R. I'm plotting R, the decay term, and in this case, WR with W less than 1. And basically the difference between these two is dr dt, and we see exponential decay. Vice versa, if WR is greater than R, then there's too much feedback and you get exponential growth. And only when W exactly equals 1 do you have an integrator. That's the fine-tuning problem. So the question is, can we modify this picture in some way that makes us not need a precise W equals 1 condition, a perfectly tuned eigenvalue, if you wish. And here's the concept. So this is actually showing a real neuron from the goldfish integrator. And you can see there's this extensive dendritic tree. And the idea is dendritic branchlets could act as bistable digital elements that flip on and off like a flip-flop. And therefore, maybe, even though we think we have analog behaviors, maybe we are more like have digital neuroelectronics and get the robustness of digital electronics out of bistable neurons or bistable dendritic compartments. And I should say, even though I'm going to show things for bistable dendritic compartments, you can instead imagine a neuron which is receiving input from many bistable other neurons that are bistable. And that is more the models that Alex Kulikoff and John Listman did. Okay, so in this case, it's more like the idea is maybe if it's more like a ratchet. So you actually get sort of held by this, held in place, and it really takes a larger kick to kick you out of it. Okay, so dendritic bistability, I just want to define what I'm talking about here. It's been observed experimentally, and it's due to the self-sustaining properties of voltage-activated inward currents, most typically, NMDA, persistent sodium, or calcium. So the idea is, here's a presynaptic input, and that presynaptic input is being ramped up, and then and as it ramps up, the y-axis showing the voltage of the dendrite. So it's ramping up the input, and then boom, here the calcium channels activate very rapidly. There's a positive feedback running to saturation. So it's like the unstable case I was talking about, until activity saturates here. And then it's self-sustaining because there's a positive feedback loop. These calcium channels, they are opened by elevated voltage, and then they let in positive ions that keep the elevated voltage so that the voltage can be maintained, and only at a much lower input rate do the calcium channels inactivate and return the firing rate to its low branch. So now, the other comment I'll make is anatomically realistic models suggest that different dendritic branches may behave approximately independently, so I'm going to use that in the model. But you could also just make a model of independent neurons providing this bistable process. So here's the architecture of the network. We're gonna have a bunch of neurons with independent bistable dendrites characterized by a function d, and firing rates characterized by a function r. So this is the function d of r. When the presynaptic input reaches a threshold r on, the dendrite flips up, and only when it returns to a lower level r off does the dendrite turn off. So it's a flip-flop. And now we're going to build the same style of model I was talking about before. Tau dr dt is minus r decay term, but now instead of a linear network or even a nonlinear network, j equals 1 to n wij inputs, the inputs are going to come through these bistable dendrites, d of rj. And then there's the external eye movement commands, that's the saccadic burst to be integrated. So, and I want to now look at, again, to have persistent activity, we need to have the decay term balance this sum of recurrent input terms. So I want to, again, do that geometry. So here, the idea is that now we can, again, plot as a function of r, the firing of a neuron, its decay, which is just minus r. So that's the green decay line. And then we can plot the inputs, sum of wij, d of rj. And imagine that the thresholds to turn these on are staggered. And in fact, in the oculomotor neural integrator, that we think could come from different background rates, and actually the neurons have different thresholds. But the idea now is, as the activity in the network increases, first dendrite 1 flips on. That's where I am here with my cursor. Then activity of the network increases more, dendrite 2 flips on. Then activity increases more, dendrite 3 flips on. But as activity decreases, now activity has to go to a much lower level before a dendrite flips off. So the feedback now forms a band, and you get fixed points as long as these rectangles corresponding to the bistable feedback loops encompass the line. So we're no longer trying to overlap a feedback line with a decay line, but rather a feedback band with a decay line. So if you take this to the limit, n goes to infinity of many, many bistable inputs, you just have a hysteretic band of stability. And as long as that encompasses the decay, and this is just supposed to be showing one of these loops, the system is stable. So one can work out analytically that the percent mistuning tolerated goes as the width of the band divided by, when plotted as a function of eye position, divided by the maximal eye position maintained. So this is just the idea. And to connect to Gordon's talk, interestingly, if things aren't perfectly tuned, then the band will shift back and forth, and that's shown here. Let me first actually show the left. if you mistune decay and feedback in a conventional model where there's no bistability, here feedback has been decreased and now the decay is greater than the feedback and one gets rapid exponential decay. Here, the feedback weights have been decreased, but now it's fine. It still maintains stability because the bistable loops are still leading to this band that encompasses the feedback line. So it's still robust, and you can see that different eye positions are still maintained. And the final thing I was going to say is if you add noise to this, then actually the decay timescale can be governed not by the timescale of sort of the deterministic dynamics, but rather by the timescale of having hopping, sort of like what Gordon was talking about when he was talking about having a washboard potential and having noise hopping, having hoppings from one well to another. It's really the stochastic hopping from one state to another that will make activity either decay or diffuse within this network, and that can be on a much longer time scale than the time scales of the intrinsic components. And that is it for that story. So if people had questions about this, the punchline in the story is we think that basically maybe the long time scale dynamics could actually come from, short time scale dynamics that work together to make flip-flops, to make bistable processes. And then you're coupling those bistable processes together to get long time scale emergent phenomena.
Good. So before we start doing anything with questions, we should all thank Mark. This was great. A tour of a large body of work.